These two sessions running the talks is to give an idea of the approaches that are being used and maybe some of the issues there are in individual groups, and those by the end of the afternoon. Really what we want to do is be able to then set the scene for tomorrow, set some discussion topics for tomorrow, and working in smaller groups, draw on the expertise that's in the room to try and see how best to move forwards on those questions. One other thing that will be interesting to discuss as well amongst the group is whether or not there's a need for a second similar meeting, where that needs to be, maybe what the meeting needs to focus on, and maybe who could host it, those sorts of things. I'm going to chair the first session, and Jonathan Cox has agreed to chair the second session, so I'll introduce myself. I'm a medical microbiologist, academic medical microbiologist at the University of Plymouth, and I'm also Chief Scientific Officer at Biologics, a company that we spun out last year to try and commercialise some of our lead compounds. So what I want to do is just give you a real overview of what we do in my group. There's a large number of the group here, and thank you to them for helping out over the weeks leading up to today and today. And then I'll give you one example of the compounds that we're working on, and I will then just pose a couple of thoughts at the end. In terms of the sort of expertise that myself and my group can bring to this sort of network, as I say, my background is in medical microbiology. Most of my postdoc research and early lectureship work at the University of Manchester and now Plymouth is in the field of medical microbiology focused on antimicrobial resistance. My group is predominantly involved in natural product discovery and development. So we try and optimize isolation methods, isolation media. We use a lot of chromatography methods to separate out compounds from bacteria grown in broth cultures. We've got pretty good access to clinical multi-drug resistant strains, and this is another sort of thing that we might want to think about as a combined resource amongst the group. So we've got good links with clinical microbiologists in Plymouth and from previous work. We carry out fairly standard in vitro tox testing with cell lines, but we also use the Galleria melanella larvae assay, which we find quite useful. The sources of samples that we recover bacteria from for the natural product screening range from environmental isolates and clinical isolates in previous work, but more recently we've been focusing on some deep sea sponges with Poppy and Matt, who are here. There's a bit of a theme for today from deep sea sponges. Growing interest in these as a source of novel organisms that produce potentially novel compounds. Because we can't recover all the bacteria in any of the samples we look at, I'm sure we're all familiar with this, and because there are cryptic loci in a number of bacteria in the environments, we carry out genome sequencing of any organisms we're interested in. We're getting into a bit of metagenome sequencing, and you'll hear more on Sam's work on that later on today. And we do in silico mining for natural products, and there's an example here where we had Abdu's at the back, had an organism again from a deep sea sponge as part of his PhD, sequenced the genome of the organism, did some in silico mining with anti-smash, predicted a locus for an antimicrobial peptide, had it synthesized and demonstrated activity, so kind of closing the loop on the genome-based discovery side of things. I'm also so most of my work is focused around antimicrobial peptides, bacteriocins, moving a little more now into secondary metabolite work. And we've got some work that I mentioned looking at rational redesign and design of antimicrobial peptides. Because we're interested in antimicrobial peptides, I'll just kind of emphasize this fact really in a way. So this is John Rex summing up after the FDA's workshop on non-traditional antimicrobial approaches in August 2018. His closing thought on summarising the workshop was that in 10 to 15 years, many non-traditional approaches will be kind of business as usual. So I think we need to think about how some of these non-traditional approaches become more integrated into the sort of the development pathway for novel antimicrobials. And as an example of some of the work that we're doing, I'll just describe epidermicin to you, which is a compound that was discovered in my group. It's produced by Staph epidermidis. It's an unmodified 51 amino acid linear bacteriocin, but it does adopt a very defined structure. We solved the crystal structure with Jeremy Derrick at University of Manchester. We've done pretty much all the in vitro characterization you would want to do with this. We demonstrate its very low toxicity, rapidly bactericidal against gram-positive bacteria, as you may expect. It's a membrane active agent. We're characterizing the mechanism of action in collaboration with a number of groups. And we're doing biophysical analysis with Max Ryadanov at the National Physical Laboratory using atomic force microscopy. In the sort of standard resistance generation methods, we can't generate resistant mutants for this compound, which is compromising our ability to look at the mode of action actually, but potentially it's good for future clinical use. And because of its properties, it's really ideally suited to topical therapy for gram-positive infections. And that's the route we're heading. So we did some evaluation of activity in Gileria melanella wax moth with MRSA. So these larvae were infected with MRSA and then treated with epidermicin, and it protected from infection. That then justified an animal model evaluation. So we put epidermicin into a nasal decolonization model of MRSA carriage in cotton rats. This is about the best model that there is for this clinical indication. You can't see the detail here, but essentially the data is showing that a single dose of epidermicin administered at the beginning of the 5-day treatment period, single dose was as effective as 3 doses twice daily, so 6 total doses of mupirocin, which is the only compound that's licensed for use in nasal decolonization of MRSA carriage. And that's unprecedented efficacy in this model, and it justified taking epidermicin further into preclinical evaluation, and that's what we're doing at Biologics. Just to sort of give an indication of the process we follow and the resources and expertise that we've got that we can kind of offer anybody that's interested in collaborating. Focusing now on marine ecology and sampling, but also sampling from many natural environments. Work with Kerry Howell, a marine biologist at the University of Plymouth. Gary Smirden is somebody who we're talking to now. The Diving Diseases Research Centre is just up the hill from us on the Plymouth campus, northern campus. As you would expect, a diving diseases centre. They've got hyperbaric chambers here, but they've also got labs where they can pressurise on a small scale microbial culture. So we're looking at trying to pressurise and manipulate the atmosphere within culture vessels to try and increase the recovery of live bacteria. Mike Allen at the University of Exeter and Plymouth Marine Lab runs the single-cell genomic facility, so we've got access to things like flow cytometry and atomic force microscopy there. I've mentioned some genomics that we're doing, and that includes Gary's work on using the MinION to try and look at metagenomes and genomes of active bacteria. Natural product characterization we do in my own group, as I say, the National Physical Laboratory. Biophysical analysis of these compounds is there's a large resource of expertise that Max is happy to kind of collaborate with people on these sorts of projects. Rational redesign, we're working on a project with the National Physical Laboratory, but also with IBM, who are using their molecular modelling tools to look at the interaction of peptides with biological membranes. That's not as promising as we hoped it would be, but it's allowing us to try and in silico design derivatives of epidermicin and see how they would interact with the membrane. A large part of what we do and what we have done over the last couple of years is working with an industrial biotechnology company in Genza up in Edinburgh, and they've been developing over the last 2 years a scalable production system for epidermicin, because clearly if it's going to be useful in the clinic we need to be able to produce large amounts of it. And they again are interested in working on similar projects with other collaborators, and anything that we we take all the way through to preclinical evaluation if it looks good throughout this whole process. These are just some of the AMR projects that we have running in the lab. So that's the RBM Ingenza rational redesign project. We were also looking at some vaccine development with a company that spun out of the University of Plymouth a couple of years ago, looking at trying to develop vaccines to reduce antibiotic use in agriculture. We're part of the One Ocean Hub, which is focusing on the marine environment, as you would expect, but we are working in a work package looking at the bioactive potential of samples from marine environments. And I've just mentioned embryological preclinical development. Just the last couple of seconds on the sort of development needs and bottlenecks within the work that we're doing. So we can isolate organisms, we can't isolate everything that's there, but we can isolate organisms that have bioactivity but then and purify, but it's the processes downstream of that really. So things like mass spec, NMR, and we're starting to work with people like Paul Race in Bristol on those sorts of things, but it's often difficult to find people with those sorts of skills that have enough of an interest and expertise in bacterial or peptide, antimicrobial peptide analysis to be able to really help. Early dereplication is an issue for a lot of people working in this field, so keen to hear other ideas about dereplication. We haven't had much of a need for medicinal chemistry yet, but it's great to have a sort of registry of people who are able to help on the medchem side. With these antimicrobial peptides being optimized or being ideal for topical delivery, we're also now keen to look at ways of trying to deliver them systemically. So through collaborators, maybe people have got ideas about systemic delivery of protein-based antimicrobials. And I also wonder whether we need to have a discussion around influencing funders like CARB-X, who for obvious reasons only focus on, at the moment, agents that are directly active against gram-negatives. Gram-positive is still a major cause, a major burden of disease, and whether that kind of conversation needs to be raised with the funders, and if there's anything that we can do around that. So just to thank Florrie and Gary and Martha for setting this up, and for the University of Plymouth for funding it, and then the funders of the research that's in the group, and for my group for being involved in all of this. And thanks for your attention.
I'm Jonathan Cox from Aston University. I am a lecturer in microbiology currently, have been for the last 3 years. And my research group at Aston are focused on mycobacterial drug discovery. So that's what we do. We're interested in the physiology of clinically relevant mycobacteria from the perspective of antibiotic resistance for the development of new antimicrobials. Before that, I was a research fellow at GlaxoSmithKline, and my focus, again at GSK in Madrid, was TB drug discovery. So running large, high throughput screens to discover new small molecule inhibitors with activity against mycobacteria that we could then look at the mechanism of action and exploit chemotherapeutically. And before that, I did my PhD at the University of Birmingham on TB drug discovery. So all in all, I've had about 10 years looking at tuberculosis and trying to find new antibiotics for TB. So with the theme of trying to identify bottlenecks, I'm going to talk you through some of the interesting challenges that I've come up against, and hopefully explain some key points in TB drug discovery that I'm sure will be relevant to wider drug discovery for antibiotics. So this is the TB drug discovery pipeline. So you start off with your initial hit compound, and you need to first identify the mechanism of action of that compound, identify the target, identify how it works. Then you can look to see if you can find more compounds that hit that target, and develop those compounds into your lead molecules that are the best possible versions of the initial inhibitor that you found, and then they go into lead optimization and into the sort of clinical pipeline. it's really important, this first step here. This is what we're really interested in, this target identification and finding other inhibitors that best hit those targets. Because case study is bedaquiline. Bedaquiline is the first new TB drug to be approved for 40 years. Bedaquiline was the result of hundreds of millions of dollars of pharmaceutical investment to develop this new inhibitor, and the pharmaceutical industry pushed this forward without spending time and due diligence thinking about the mechanism of action of bedaquiline. Bedaquiline was approved to treat multi-drug resistant TB and extensively drug resistant TB, so if you're one of the unfortunate few that gets that infection, there's a chance that you will be prescribed bedaquiline as part of your treatment. If you do survive the infection and the treatment, you'll end up with a pacemaker for the rest of your life. Because bedaquiline, we subsequently found out once it was approved, inhibits the ATP synthase, which is a highly conserved protein. And so, although it is better at inhibiting the bacterial one, it still is very toxic and has some very nasty side effects in people. So that is case in point why it's so important to know how your antibiotics work, what they target in the bacteria, to make sure that we don't have the same thing for them to target. So this is sort of the classical dogma of drug discovery, of antimicrobial drug discovery. There are two schools of thought. You either take the target to drug strategy, where you have your drug target in mind, you know what it is you're trying to actually inhibit, and so you design your drug to see whether or not you can actually get activity and have the drug penetrate the bacteria, get in and selectively inhibit that target. That's the target to drug strategy. And the drug to target strategy is where you start off with your molecule. You've done a big screen, say, of thousands of different small molecule inhibitors. You've found the ones that have activity, and then as a result of that, identifying that molecule, you then identify a new target and improve and refine on that target. So two different strategies. And I'm going to give some examples of work I've done for each of these strategies and some of the limiting factors with each of these strategies and things to consider. So I'm going to start off with the drug to target approach. So first of all, you conduct your high throughput phenotypic screen of 100,000 small new chemicals, new small molecules, to see which ones actually have activity against your organism, which in my case is Mycobacterium tuberculosis. You then rank those inhibitors that you've identified, and to explore them further, the process we used is to generate spontaneous resistant mutants against those inhibitors. So you force evolution, artificially high concentrations of drug to very high sort of bacterial load to select for a resistant phenotype in the organism that you're testing. So you generate your resistant mutants, you then sequence, first of all, you've got to validate that resistant phenotype, make sure it's not just a side effect of the screening process, and then sequence the genome of those resistant isolates. And then using bioinformatics and identifying the single nucleotide polymorphisms, you can see where the resistance comparing mutations are, and if you're lucky, those resistance comparing mutations appear within the gene that expresses your target protein. Okay, so that there's a mutation that changes the target protein sufficiently to cause antibiotic resistance, and that gives you then your smoking gun. You can then validate genetically. Okay, so you can overexpress, which is one of the things that I've done a lot in my career, is we use overexpression to actually show that when you produce increased levels of the target, that you get a shift in the inhibitory concentration, you get sort of induced resistance in order to validate target engagement in the cell, and then onwards to further mechanism of action studies on that point. But one of the big problems with high throughput screening is that the main message is that a high throughput screen is only as good as the library you're screening. And in 2014, I think it is, it's just cut off there, yeah, 2014. This paper was published in Nature that talks about the PAINS compounds, which are pan-assay interference compounds. So what these molecules are, effectively, is highly promiscuous chemical entities that will very effectively kill bacteria in phenotypic screens, and they are something of a red herring. And many people have spent a lot of time and a lot of effort and a lot of money and a lot of energy in their groups exploring their new hit compound, which actually is one of these pan-assay interference compounds that is highly reactive, and so it will never make it through into sort of preclinical drug discovery. So this is a major reason for the high attrition rate in small molecule drug discovery taking this strategy. So the PAINS compounds are something to be aware of there. So if you end up pursuing your hit molecule and you take it through and you generate your resistant mutants and you get your whole genome sequencing data and you find SNPs that look like they're in the perfect protein, you have to be really careful. So this paper was published in 2012 that states that these new compounds, which I'll abbreviate to the THPPs, target a protein in TB called MMPL3, Mycobacterial Membrane Protein Large. They found SNPs within MMPL3 that appeared to be the resistance-conferring mutations, and so assumed that that was the target. And then in 2016, myself and colleagues published this work with the same compounds where we'd actually taken a different strategy beyond the whole resistant mutant generation, whole genome sequencing approach. We'd done a bioinformatics-based strategy, a pull-down experiment immobilizing the compound using clarified lysate of bacteria and pulling out the target that we actually found that bound directly to the compound and identified it by mass spec, and then went through a whole long, very extensive process of validating this as being the true target, and that MMPL3 was in fact one of those red herrings. It's likely to be the means by which the antibiotic actually gets into the cell rather than being the true target. So the point I'm making here is you have to not believe every bit of evidence that you get, particularly with whole genome sequencing data. It's a guide. It can help you to narrow the field down, but it's not everything. And if you publish it as everything, then invariably someone else will come along and publish the alternative answer. So whole genome sequencing has limitations. It sometimes gives you a hint. It's not the only strategy, and taking a biochemical strategy, a phenotypic strategy, is always a strong way to go. And HTS is only as good as the library that you're using. So the target to drug approach then. Well, what we did was I worked with EMBL, and using the GSK small molecule library, EMBL got the crystal structure of dihydrofolate reductase, which is a well-known target in TB, and in silico screened the entire 4 million compound library that GSK had, and all the structures, so did this all in silico, identified these new inhibitors, which we then tested biologically and demonstrated that they did have whole cell activity and that they did inhibit dihydrofolate reductase. So that was a completely different strategy. We'd found these small molecule inhibitors by focusing on the target. But the challenge is with this approach is you don't know that the drugs are actually going to get into the bacteria in the first place. If you focus too much on the target, you don't know whether they're going to have whole cell activity. TB has a big cell wall that these inhibitors have got to get through. Okay? Are they going to be active? Are they going to be effective against the organism? Do they engage the target, or do they get in and just do lots of different things to kill the bacteria? And the other big problem with this is that you'll never discover a new drug target because you're looking at the same old drug targets. So what about trying to think about something that covers the good aspects of both of the dogmas. So we took, this is my last slide, we took this approach to have a combined strategy whereby you identify key proteins, important targets, or likely useful drug targets within the organism, and then we engineered overexpressing strains for each one of these targets and screened them against 10,000 known inhibitors of TB. And what we were looking for was an IC50 shift, a resistant phenotype upon overexpression of the target to indicate that that could potentially be the mechanism of action, and that we had whole cell activity, and that we had target engagement. And we did this, we identified 284 new hits, and the take-home message is here, we're still publishing papers on it many years later, because we generated such a huge pool of compounds with a highly suspected mechanism of action. It gave a really solid starting point for a lot of work. Okay, so it's combined two strategies. You get new drug targets, you get good inhibitors because you've removed all of your pains compounds, you know you've got whole cell activity, and you also know that you've got target engagement. So it's this slight change in strategy that enabled us to do drug discovery, small molecule drug discovery, that was really likely to generate things that we can move forward with. 12 minutes. Sorry about that. Thank you.
We've got time for one quick question.
Just a quick comment on the last slide.
Sure.
If you're,
I raced through it.
It doesn't matter. You don't have to go to it.
Okay.
If your compound had been fluoroquinolone, overexpressing target would have the opposite effect, namely increased sensitivity, not resistance. So would you not pick it up by your estimate?
We would still pick it up. Absolutely. So we looked at shifts in both directions, and explored everything from, absolutely. Yeah. It wasn't just the resistance. I was trying to sort of, simplify the theory behind it.
Thank you very much. Okay, thank you, Jonathan. Leading straight into Tony Maxwell from John Innes Centre. Thank you, Tony.
Great. Thanks very much, and thanks to the organisers for organising this meeting in such a lovely place, albeit difficult to get to, but I'm sure everyone's going to say that. So I'm going to give a brief overview of what is going on in my lab, which is work on topoisomerases and how they can be targeted for antibacterial chemotherapy. So we work on a range of different topoisomerase enzymes, mostly from bacteria, so gyrase and topo4s, particularly from a range of gram-positive, gram-negative bacteria, but also enzymes from plasmodium and plants. And we're interested in a whole range of topoisomerase relevant drugs and toxins which are listed there. And what we have is a range of different tools, including biochemistry, molecular biology, structural tools. These are all in-house, plus a number of other approaches which we do by collaboration. We also have an interest in natural products per se, thinking about novel plant toxins, and as a previous speaker has said, we have Galleria set up as a test system to look at this. What are we looking for? Two things really. One is fundamental knowledge about these enzymes, their structure, mechanism, and physiological role, but moreover, and more relevant to this meeting, understanding drug action resistance and development of novel antibiotics. So one slide on the biology. I should say that some of the work is done in conjunction with our spin-out company, Inspiralis. So a number of biological processes in cells lead to topological problems in DNA, specifically DNA replication and transcription can lead to unwanted supercoiling or interlinking and catenating of DNA. So these problems are addressed by the topoisomerases, and there are essentially two types. Type 1 break one strand of the DNA transiently, and type 2 transiently break both strands of the DNA. And as of now, there are currently no antibiotics targeted to type 1 topoisomerases. That's a discussion point for the future. They're all targeted to the type 2 enzymes, that's specifically gyrase and topo4, both of which have been highly successful antibiotic targets. And indeed, in the work on TB, we just talked about the moxifloxacin and gatifloxacin target TB gyres. So why? Well, the enzyme has quite a dynamic mode of action involving a lot of conformational changes, and I'm not going to go through it in any detail, just to say you have two subunits which combine together to make this nanomachine on DNA, which then does these gymnastics in terms of breaking DNA, grabbing another piece of DNA and passing it through. So this is the generic mechanism of gyres, but it really accommodates other type 2 topoisomerases such as Topo4, which do this DNA breakage, strand passage and reunion reaction involving a lot of conformational changes in the protein, and we think that's what makes these enzymes such great targets, because it reveals hydrophobic patches and so on and so forth, which can be exploited by small molecule antibiotics, like the fluoroquinolones, which interrupt the enzyme at the DNA breakage reunion part and actually lead to double-strand breaks in bacteria, which is a very effective way of killing cells. Aminocoumarins like novobiocin, which block ATP binding, and one of the newer compounds, simocyclinone, which block the binding of DNA to the enzyme. So our approach has been biochemical, biophysical, structural biology, such that we can build up a 3D model of the enzyme and show where different compounds, fluoroquinolones I've talked about, bind here where DNA would bind, so does simocyclinone. Aminocoumarins bind where ATP would bind, and thiophene are an example of a novel group of compounds that we're working on, which bind to a completely unexploited pocket for which there's no cross resistance. So our basic thrust is the molecular basis of compounds which act on this and related targets. Okay. So how do we do this? We have a variety of different funding and collaborations which do this. So in terms of EU funding, we are a member of the ENABLE consortium, which is one of the Innovative Medicine Initiative consortia, the so-called New Drugs for Bad Bugs, and that's specifically targeted at new compounds against gram-negatives, and that's got 30 partners, including GSK and Sanofi. We were also a member of the More Medicines for Tuberculosis consortium, which has now finished, but happily they have 2 drugs in clinical trials, not against our target, against another target, and it's an example that these things can actually work. I oversee a group on the Norwich Research Park, which includes Sainsbury Lab, it includes hospital, university and so on, called Antimicrobial Research on the NLP, which is about 40 labs working in different diverse areas of AMR, and we're able to provide reagents and screening services mainly through the spin-out company Inspiralis. And I also serve on the scientific advisory board of the charity Antibiotic Action, which is a public information body which tries to raise awareness of AMR, particularly amongst politicians, and it's been quite successful in doing that. We were asked to identify bottlenecks, and I think it causes me to say that what we do is a very small slice of a very big cake. And I think that's true for a lot of people in this room. What we are actually working on is not going to achieve novel drug discovery in of itself, but it's part of a much bigger picture. So what do we need? We need new compounds. And I think the main problem there that we find is we get the same old, and we need to get into new chemical space, much more diverse compounds. The same old targets come back again and again, and gyres and Topo4 are such example. And what we do need, I think, is new chemistry. We also need funding at an appropriate scale. And it's fine to get sort of low-level funding from BPSRC and Wellcome Trust and so on, but I think what this area needs is actually big money putting into it, and this has to be done sooner rather than later. And finally, and this meeting is a good manifestation of this, we need ready access to good collaborators, people who can do the sort of things that we need to do. But these are likely to be in the academic area rather than in the big pharma area, in my opinion. And I just want to say two more things. One is an advertisement that I'm editing a special issue of JMB, Journal of Molecular Biology, on the molecular basis of antibiotic action and resistance, which is coming out in the next few weeks, and there's lots of very relevant articles in that. And finally, I'm not going to discuss this, but in the past we've been challenged to come up with a pipeline for antibiotic development really based upon academic expertise, and this has been put together. There's too much on it to take you through this, but basically how you can go from what's called compound equity to new drugs. And I think it is feasible now to do this using academic expertise and small companies. Oh, that's only 8 minutes, that's great. Okay, so thank you for your attention.
Perfect, thank you. And I think what you're saying there at the end really gets to the heart of what ABX is trying to do. So thank you for that. Yeah. Any questions?
Yeah,
there's a few questions. Firstly, so I'll just get questions out first. So with DNA topoisomerases, you've got two groups, 1 and 2. Within those groups, you have different classes, different types.
correct.
Okay, so presumably in bacteria, then you may have. Are they all the same type in bacteria?
Yes.
Okay. Well, that's the question I was going to ask. Do you think it could be that the bacteria may produce compounds that will inhibit the bacterial Topoisomerases?
They do.
They do that.
They do that, yes. And so they defend themselves. So one good example actually relates to Matt's talk, that microbial toxins are produced by bacteria to kill other bacteria, but they defend themselves against it by producing antitoxins or ways to get around. So yes, they do exactly do that.
And they inhibit the Topoisomerase?
Absolutely, yeah.
Okay, and then finally, you talk about coumarin and plant-derived compounds. Is it coincidental that they're active against these molecules, or are they an evolutionary interaction?
So novobiosin, which is sort of the most well-known aminocoumarin, is a Streptomyces natural product, which is produced by Streptomyces species. When that operon is turned on, it makes a novel Jarvis B subunit which contains a resistance mutation. So the Streptomyces defends itself by making its own resistant subunit, but then it kills all its friends and neighbours in the soil. So that's a good example of how you can make a lethal molecule but not kill yourself.
Yeah. All goes on, does it? All goes on.
Any other questions? Comments? Okay, thank you. So I think we'll move straight on to Catherine back from University of Bristol.
Okay, great. So good afternoon. Yes, as Matt said, my name is Catherine Back and I'm a PDRA at the University of Bristol. I'm just going to give you an overview of what we've been doing in our research group and also describe some of the bottlenecks that we've had issues with. So our project is bioprospecting for novel natural product-based antibiotics from deep sea sponges. Oh dear. That's annoying. Let's see, something's happened.
You made your talk on a Mac.
Yeah.
And the Mac doesn't like it.
It's not a Mac though.
Yeah, it should be okay.
Okay, well, we'll have to just go with it. Okay, so why are we bioprospecting from sponges? Well, they are a really old organism. They're thought to be the first multicellular organism on the planet and consequently are the common ancestor of all animals, or metazoa. They've been around for 640 million years and they have a diverse community of microorganisms that exist within the structure of the sponge, containing bacteria, archaea and fungi, which exist symbiotically or commensally with the sponge and generate nutrients for the sponge to use. They are known, these species are known to be some of the richest producers of bioactive natural product metabolites and are consequently used by the sponge for protection against more pathogenic strains. and a previously untapped resource for novel antimicrobial natural products. Is this going to work? Yes. Okay, so our collaborator, Dr. Kate Hendry in the Earth Sciences Department, has collected a wide variety of sponge samples from the mid-Atlantic between West Africa and Trinidad. And I don't know if you can see on this map, but you can see the stars are the sampling places. She uses a remotely operated vehicle, or ROV, deployed at depths between 0.5 and 4 kilometres to pick the sponges off the seabed, as you can see here in this video. Okay, so our method for identifying strains that are producing antibiotics and then attempting to isolate the compounds that are the antibiotics is as follows. So we homogenize a sponge sample using just a pestle and mortar. We spread the homogena on a wide variety of different agars to hopefully isolate the widest variety and the largest number of strains as possible. We then isolate colonies to pure culture and test them for antibiotic activity. We use a soft agar overlay assay, as you can see here, where we grow the strains on the agar that they were isolated on, and then use a soft agar containing a pathogen which we pour over the top and leave overnight at 37 degrees. And if the strains have activity, they'll have a zone of inhibition around them. We can then take that strain, grow it in liquid culture, hopefully, and then we extract compounds from the supernatant of the culture media, run it on an LC-MS, and hopefully identify any compounds that are within that extract. We can then purify them and use NMR at the university to identify the structure of the compound. We also use 16S RNA sequencing to identify the strain, and if the strain is producing something interesting, we can use whole genome sequencing, so we use a combination of Illumina and nanopore sequencing to identify potential biosynthetic gene clusters. So the bottlenecks that we've been experiencing, well, we've had several, but these are the two main ones. So we have hundreds of sponge samples in our freezer, and there are just way too many to process for just the few of us who are working on it. I think Sam's going to be talking about that bottleneck in the next talk. But also, out of these hundreds of sponge samples, we can only really pick out about 5%, roughly 5% of species that are within this environmental sample, just using our agar plate assays. So we'd like to address that. We're also having trouble when strains lose activity when they're grown in liquid compared to on an agar plate. And we're hoping to develop methods for growing strains, particularly these actinomyces and streptomyces, to produce as many compounds as possible and as much compound as possible within a liquid culture, as it's the easiest way of extracting out these compounds. So one thing we've been using or developing to try to increase the yield and diversity of strains to go from this 5% strain isolation and pick out at least some of this 95% of microbial dark matter which we're unable to culture. We're using the isolation chip, or iChip, which was initially designed by Slava Epstein at Northeastern University, and he has shown that it has increased the number of isolated colonies of up to 50% instead of 5%, which is great, and also increased the number of novel strains, which is really important. So the method is as follows. So he used soil, obviously we'd be looking at sponge. They took soil samples, dilated the soil sample to a particular concentration of microbial cells, and suspended in a soft agar, you can pour over the top of the central part of the iChip, hopefully to get about 1 cell per hole within the iChip, so around about 1 cell per microliter of agar. You then sandwich this between semi-permeable membranes to allow passage of nutrients and secondary metabolites, but prevent any passage of microorganisms. the eye chip is then clamped together and screwed shut, and you can then put the eye chip back into the environment in which you found the initial sample, allowing the bacteria to grow within conditions that they would normally be growing in. So we have 3D print files from Northeastern University who sent to us from Amit Pinto. We have printed them, and we've been attempting to sort of develop a method that works for us. Unfortunately, we've been having trouble with the sterility of the eye chip, because the plastic we're using isn't autoclavable, unfortunately, and even using ethanol to sterilise it, you can't leave the ethanol on too long because it starts to warp the plastic as well. So yeah, we've kind of stopped using the small, I think the battery's running up, the small eye chip, and we started looking at the larger eye chip, which is described in this paper here, Nature Protocols, which is essentially a pipette tip holder, which is thus autoclavable, which is great. It's much bigger, it's about that big, like a 96 well plate, and you do the same thing, put your bacteria into soft agar, and then you can pipette into the holes and let it set, and then stick semi-permeable membrane on either side with glue this time to make sure it's completely contained within each plug. We're hoping to develop that soon. I'm just waiting on the membranes, which have not arrived yet. So I'd just like to thank Kate Hendry, who took our sponge samples from the Atlantic, the Bristol Genomics facility for doing the Illumina sequencing for us, Gilda and Gary for helping us with the nanopore sequencing, Jim Spencer, who's given us access to Cap2 space, and also Amit Pinto for sending us the iChip 3D print files. And thank you for listening.
Thank you very much. Any questions, Jonathan?
I've got two super quick questions. One, UV sterilisation, would that work for your plastic?
Oh, potentially.
Okay,
I think there was a reason why we didn't, but I can't remember
The light
exactly.
will get into all the nooks and crannies.
Yeah,
Does it not?
I think it would be a little difficult, but yeah.
The other thing I wondered, the difficulties with culturing the bacteria, would there be something in trying a sponge homogena within the agar as a kind of a supplement, a growth supplement?
So we have been using
the
that.
environment. Does that work?
So we've done a few extractions and used this. So sponges are made up of spicules, which are made out of glass essentially. So we've done spicular extractions and then added that to the agar as well, and it has helped actually, I think. Yeah.
We've done aqueous and organic
Yes,
extractions as well.
Okay. To see if you find that magic ingredient.
Yeah,
That's great.
It's a very good one. So you with the iChip you can capture 45% more diversity.
Potentially.
So but you isolated 45% more normal compounds then?
Maybe.
Well, have we?
Oh, have we?
Yeah, how successful?
Oh, we like I said, we've been having issues with sterility with the iChip. We're just really developing a method at the moment. We haven't actually been able to use it for actual identification of new species yet.
Okay, thanks.
Are you getting a sponge mush and putting the iChip on that mush, on the sponge? How
So-
would you plan to use it?
Potentially, that would be something we might be able to do. At the moment, we've literally just been using seawater in the cold room. So, but I think it would be cool to be able to perhaps put it back in the sea somehow, or at least a sort of more I don't know. Yeah, potentially alongside a sponge, maybe in an aquarium or something, would be really good.
With the initial experiments where you were isolating and separating colonies to look for antimicrobial properties, did you ever combine different species to see if they were producing other compounds when in a combined environment?
So yeah, that's something Sam's been doing. Yeah.
Yeah, a little.
But with more pathogens. Yeah, combining and
not
seeing.
using two sponge species together, but kind of using the marine ones we're getting with kind of like other pathogens and growing them together.
But I think that's something we would be interested in doing in the future. It's a little difficult, complicated thing to do. But yeah.
Do you think pressure is a factor to the growth?
Yes, probably. But as these guys probably know, it's difficult to culture bacteria at high pressure, and then being able to then make large amounts of, or grow large amounts of the bacteria at high pressure. So it's all very well growing on one plate or one small culture, but if we want to scale it up, it becomes a little bit more difficult. But maybe these guys have got the answer.
I think we're really intrigued to see what we do find when we work with DDRC, the Darwin Diseases Centre. And it may be that you can start to domesticate in the same way that the iChip does. You start to domesticate the organism so they then are able to grow outside that pressured environment. But whether that changes their metabolism and they switch off lots of these secondary metabolites, I don't know. One last question at that.
Is there any evidence for anaerobes or streamophiles such as archaea in symbiosis of these sponges? Not just isolated with the sponge, but actually grown inside.
Do you mean grown inside the cells?
Yeah.
I'm not sure. Too much work has been done on actually looking where the bacteria are existing within the sponge. We kind of assume that maybe some of them are attached to the spicules, attached to the cells, but also possibly also inside the cells as well. But yeah, not much work has been done. We're looking at anaerobes as well. We've got some candle jars. We've been trying to isolate anaerobes as well, but haven't had a lot of success actually. We've had more hits from aerobic strains.
Okay, great, thank you very much. And so I'll lead straight into Sam, who's also from Bristol.
Okay, yeah, great. Hi everyone, my name's Sam. I'm from the University of Bristol, working on the sponge project as well with Catherine. So what are we talking about is possibly how we can kind of navigate one of these bottlenecks of having far more samples than we can ever process by kind of using a metagenomic 16S guided approach to kind of pick samples before we start culturing them. Because I think a lot of the time in natural product discovery, we're kind of dealing with a bit of a needle in the haystack kind of scenario, and some of those needles are more colourful than others, so people tend to find them again and again. So just kind of any way we can kind of intelligently guide ourselves, because we know there's more antibiotics out there in the natural world. It's just a case of kind of having the know how to find them. So yeah, some of the deep sea sponges we're working with. So the bottlenecks, yeah, there's more samples than we can actually process. I think we've got around about 100 or 150 sponge samples at Bristol, and we just don't have the manpower or the space to do anything with that really, which is unfortunate because there's always a chance that the next sponge is going to hold that kind of like really stellar antibiotic producing bacteria. So a solution that I've kind of been working towards at the moment is, can we kind of use culture independent techniques to kind of assess the microbial composition before we go to that step of culturing all the sponge on the agar, and maybe kind of make a more intelligent decision about what we're going to use. And kind of what this will give us is kind of the abundance of the microbial load on the sponge. So maybe a certain sample has 10 times as much bacteria than another one, which would be a very good one to go for. Diversity, which is obviously key when you're trying to find diverse natural products, and to some extent as well novelty. Novel bacteria are hopefully going to give us our novel natural products. So kind of the hypothesis that we're testing here then is that the diversity and abundance that I can get through the metagenomic approaches have some kind of bearing on the diversity and abundance that we can get when we're culturing. The kind of streamlined approach we've got at the moment is we follow the Earth Microbiome Project, which is also the Sponge Microbiome Project as well, the standards they use. We use the 16S primers here, sequenced by Illumina, and following that we analyse the data using CHIME and Data2 to kind of denoise all of the samples and try and really pin down what kind of taxonomy we're getting. So the reason why we're kind of using this metagenomic approach to kind of inform our culture-dependent approach, rather than just kind of maybe going for a whole genome meta approach, which definitely has some utility, is just kind of based on historical factors where the culture-dependent approach is still giving us new novel natural products after all this time. So we're not trying to reinvent the wheel here. We know we can get results with a culture-dependent approach. It's just maybe using the kind of new techniques that are coming through to kind of more intelligently guide what we do in the lab. Because once you have actually grown that producing strain in the lab and you can get that compound in liquid, it's 100 times easier to kind of take that forward. So here's some data that I've got back from this. So this is a 16S analysis of 24 sponge samples that we hadn't yet processed. So kind of just as a crude analogy here, I had a look at the phylum level distribution of actinobacteria reads present in all these sponges. So you've got the sponge sample here, kind of the one beginning with B, and then this is the percentage of reads from the phylum Actinobacteria, which are a phylum of interest to us, that were present in all those different samples. So hopefully you can see fairly quickly that 2 out of these 24 samples had almost 40% of the Actinobacteria reads present in them over all of these 24 samples. So straight away, this looks like something that would be of interest to us. Instead of, processing those 12 sponges to hit that one that's got the really high burden of Actinobacteria and possibly Actinomycetes, you can be much more intelligent with the way you're using the time and your way you're using your resources. You can then have a look a bit deeper, so you can have a look at each different sample individually. Again, this is only broken down to the phyla level. You can go much deeper, and that will be something I'll kind of be looking forward to doing. But you can get abundance, and so this, for example, this sample here, you've got the number of Actinobacteria reads here. What I'll be looking to do then is probably taking that little red slice and going further, possibly down to the family level. So then you can compare one sample to another. While it may have a high level of Actinobacteria, how much diversity is actually in that Actinobacteria that we're looking at. And yeah, so future objectives really is to test my hypothesis and hopefully start getting some interesting results, novel bacteria and novel secondary metabolites, and to really assess the usefulness of the 16S data. So I'll be doing kind of a pilot study, I guess, kind of using a limited number of media, maybe 5, with a focus on Actinomycetes. So if anyone has got any media they use for marine Actinomycetes, I'd love to hear that. And maybe take the 2 really dominant ones that have a lot of Actinobacteria in them, and another sample that has a very low number, just to kind of test this hypothesis that the culture-independent stuff you are getting on the computer is useful in the real world, and you can use it to guide your sample selection and save yourself a lot of time and money. And obviously, with the ultimate goal, which is why we're all here, is to isolate and characterize novel compounds from novel bacteria. Okay, thank you very much. I'd like to thank everyone at Bristol from multiple departments, chemistry and life science included, and by funding as well from the Medical Research Council. Thank you.
Thank you, Sam. On the timing. Any questions?
Are you planning to see how good your cultivation approaches are compared to your the more sort of non-culture bases you do? So, knowing a lot of the time, especially with that kind of bacteria, you do an approach like this and say there's lots in there, and then you lift your plate and you pick 5 or 6 things and identify them and go, it's not very good. But we did something similar to that, when we melt the agar of the culture plate and put it in, it's not that bad. I think it's just a human selection of what's being picked off the plates, like you basically inferred what you sort of referred to. Sorry. Sometimes I think it seems that we sequence everything and go, it's very diverse, and then we pick a few things from the culture plate, which is not very diverse.
Because we like the colour or they look a bit funny.
Yeah, or because 2 streptomyces are identical.
Yeah.
But you then go and sequence the genomes of the chalk and cheese. You just don't pick them because you go, I've already picked that one. So I think it'd be interesting to compare exactly what you're doing with sponges, because they're sort of not like soil hyper-diverse environments, with the cultivation techniques, and actually see how good they are.
Yeah, I think that's a really good point, and I think that's something we'll definitely be looking at, because we have been kind of, when we haven't had this data, assessing how good all the different media is and what we're covering and stuff. But until you have this kind of data, you're not really comparing them in a fair way. If there's a sponge with like 5 times as much abundance, and you're saying, oh, that was worse than that one, so we did like bad media selection. So yeah, we're going to try and keep it as stringent as possible. Like I said, take 2 with really high levels of that and 1 with a low, and kind of see where we are with that. An important thing to say as well is kind of what I'm not trying to do is to find like a species or something on the kind of culture independent stuff and say, right, I'm going to culture that. That's the one I want. What I'm generally looking for is it's got high abundance, high diversity, and kind of a level of novelty. So more of like a rough guide rather than really targeting. But yeah, it's a good point.
So like focusing on finding particular sponges that are possibly worthwhile exploring. But I've worked on sponges as well. Like one of the biggest issues with deep sea sponge biology is so many of these sponges have no IDs to them.
Yeah.
So you know, having a sponge and knowing that community is great, but how do you transplant that into, you know, where you would refine that sponge, or where you could, or even if that sponge has ever been identified before. So like, how do you think that could be incorporated into your workflow?
Yeah, I mean, so we don't really have that kind of like marine biology expertise at Bristol, which is definitely something we could work with you guys on. But I know the people who collected, we sent, I think 22 of the 24 samples they're currently in Norway there's someone called Joanna, I'm not sure if you're familiar with her. So she's doing species identification on them. I guess for the purposes of our kind of what we're doing it for, it doesn't really matter if we can go back to that exact point and find that exact species and everything. As long as we get that kind of killer producer, then we're kind of happy, and we frozen stock it 5 times so we never lose it. Have I got time for one more?
You have got time.
Okay. I tried to be very concise.
I've just got a question. I could be completely wrong, but it's a different line of thought. I think what you did absolutely brilliant, completely logical. But when we flip the question the other way around, what are the most abundant species that are in that population? What are the least abundant? Right, so when they got some sort of lethal weapon or maybe a new chemical which you're not even looking for potentially.
Yeah.
I don't know if it's a way of being an Iceland.
Yeah, I think, that's a good point. Again, it's kind of like a crude fingerprint, really. So you're not going to get that kind of functional data, like you said, that one of these really underrepresented ones have got that killer kind of mode.
Because obviously acting in bacteria are obviously very well studied. It's one of the things in those data sets you go whether there's anything else you can dig out from the least abundant.
Yeah, possibly. I think that's something over the next month or so when I'm finally selecting my final 2, I'm going to kind of dig down a bit further to kind of the family level and all of that. And I think it's important to say that while I kind of use the Actinobacteria final breakdown as kind of example of the utility of it, that's not the only factor I'm going to use it. So if there's a lot of kind of unassigned reads there that just don't have any matches in the database, that's also of interest. If there's a massive level of diversity, even if Actinobacteria maybe isn't a massive part of that again is of interest as well. But yeah.
I think we've kind of discussed this in the past in the lab. It's about the different types of sponges and not knowing what they are. So presumably they're all ancient. Presumably some of them are better complements to mediate equals to the others. Okay, some of the more advanced sponges.
Yeah.
So the more basic ones are probably better. So they may be relying more on bacteria than dinosaurs. So it's probably a good idea to know what they are.
Yeah. We're waiting. But I think that's being done at the moment. So that'll be a nice complement as well to this work, because we can match those all up.
How evolutionary they are, what bacteria are usable.
Yeah, that's a good point. Thanks.
Okay, cool. Right, I think we'll wrap it up there. Sorry, I should thanks a lot. Thank you for questions.
Firstly, I'd like to thank you guys for setting this up. I think it's going to be a very useful forum. I don't have 10 minutes worth of material, so I'm going to talk about somebody else before I talk about myself. So I'm a quarter of the microbiology team up in Antibiotics in Cambridge. We have Dr. Claire Arich, who's looking at natural products, and on the topic of the iChip, looking at how you can develop methods of growing unculturable materials and extracting different things from that. So we're all looking for collaborations, so that's possibly someone we can talk to on that. We've got Dr. Helen McCroby, who is looking at combinations of different pathogens together and looking at the genetics within them to see what changes with shielding effects and different environments. And we have Dr. Carrie Walker, who's here today as well, who's very much looking at surveillance of the passing of the different antimicrobial strains, in particular in Salmonella, and how it's spread through bird feces. So if anyone has any interest in bird poo, we can talk about that later. I'm very much interested in a nanoscience approach to creating novel ways of tackling antimicrobial resistance. And I'm going to focus then on the DNA quadruplexes today. A lot of people ask, what are quadruplexes? I get a lot of blank faces when I mention them. So these are 4-stranded DNA structures, and a couple of rudimentary cartoons of them here. So we have G-rich sequences, and our guanines are forming tetrads through hydrogen bonding with themselves, rather than the cytosines that they would normally do. So having a linear sequence of DNA, you have some G-rich regions, and they form these knot-like structures which can then interact with proteins. We have these different loop sequences,
Depending on the loop's type and formations, we can get different specific interactions with proteins. Now, one of the drawbacks of this is we don't know yet how our proteins are going to be specifically interacting with them. We can rationally design these structures. We know from looking at the sequence what loops are going to form, how big they're going to be, whether they're downside, whether they're across the top, etc. But we don't know yet what proteins are going to be interacting. So it's that mechanism of action that we need to look at further. They have been shown to form in vivo and in vitro. Very difficult to see them in vivo though, and there's still a lot of controversy over whether they actually form, but they actually do. There is more and more evidence growing that they're involved in gene regulation. So within human cells, they're all through the telomeres, so they're holding on to genetic stability, but also in a lot of promoter regions to up and down regulate genes, but also then in transcribed regions being transcribed into mRNA and producing RNA quadruplexes with different functions within the cell. Just to then try and visualize how that might impact, we've got a few different scenarios in which quadruplexes might up-regulate, down-regulate and prevent or help the transcription. So we might have quadruplexes formed on the antisense strand, which allows then the sense strand to be unbound and allow transcription to progress. On the other side then, we might have that on the sense strand and block then the DNA polymerase as it moves across, because they're stable structures and are mostly enzymatic resistant. Alternatively, we can have our protein binding, depending on the formation of these loops, and allowing transcription factors to come into play and promote transcription, and alternatively bind other proteins that are going to block our transcription factors and therefore downregulate transcription. Okay, so we know that the sequences are there, we know they can form, but we don't know what action they're going to take. So one of the drawbacks is that they're throughout the genome. There's G-rich sequences everywhere. You take any gene, you pop through a piece of bioinformatics software, it will highlight the G-rich regions and it'll exactly tell you whether they're likely to form or not likely to form. You don't know until you test it, but there are hundreds you can go through, not knowing if they're going to form, or if they form, whether they have any impact. So just to give you one example, I've taken one sequence here out of so the ADEB Efflux-Pump of Acinetobacter baumannii. Taking that series of genes, there are 40 different sequences within that in 5 different areas. So taking one sequence, I haven't put the sequence on here at the minute, but using different assembly techniques, usually through dialysis of monovalent cations. It's a self-assembly process. This here is a DNase assay. So we have DNase that will destroy all forms of DNA except for quadruplexes because they're enzymatically resistant. So we take our single-stranded DNA control, our DNase is digesting that, but at the same time our quadruplex stays stable. And we do what's known as the TDS, a thermal desorption spectroscopy. We take the folded quadruplex, measure it at 20 degrees using UV vis, melt it at 95 and take the difference. So what we're looking for there is our negative 295 peak and a couple of peaks, maybe 245, 265. These change depending on what our structure actually is. So we know then that this is a quadruplex in the assembly conditions that we have. We test it in a range of different assembly conditions. We don't know then whether it's going to form a quadruplex in cell. But if we go back to this picture here, if we know that the quadruplex in cell binds to a protein and prevents transcription, we can potentially flood the cell with formed quadruplexes, act as a competitor of this binding molecule and therefore upregulate transcription, or in this state steal all the transcription factors and downregulate transcription. So in this case we took our Acinetobacter, we popped the quadruplex in it for about a day and then done a simple disk diffusion assay to see it, and we did then see a decrease in resistance in this one in particular. Now, this is not significant in any way, shape, or form, but it does show that there's merit in progressing this and looking at all the different compounds that can come from it. So at Emily Ruskin, we are very much a teaching university, and we don't get an awful lot of time and a very, very little amount of funding. So that's my big bottleneck to research. But in terms of this particular project, we have a very slow process. There's a lot of targets, and I'm chipping through them very, very slowly. So that's not going to be a big help. In terms then of structural analysis, we want to know exactly what the structure of the quadruplex are. So I am working with a couple of collaborators looking at getting the NMR solution structure completed on those that have been formed. But also CD is a circular dichroism is a very quick and easy technique that tells us whether the strands are parallel or anti-parallel, and can help us then down the line work out what proteins are going to be involved in it. And finally then, as Jonathan said, it's very important to know about the mechanistic actions that are going on. So we need to know what proteins actually are involved. And if we can use the quadruplex to determine the mechanism of the quadruplex in vivo, we can then use the proteins that are involved in that to look at alternative treatments if a quadruplex then isn't an option or a viable option. And that's all I have.
So I was wondering, when you did your Cornwall assay, what kind of conditions did you use? Were they kind of relevant to the inside of the cell?
Is for the resistance one or the structural characterization? So this in particular one is potassium chloride. So as close as we can get, but it's very, very difficult to test whether they form inside the cell. One method that we can use is molecular beacons. So we put fluorophores in the end of the sequence, put that inside the cell, and then see if we're getting a change in fluorescence. But then we also don't know if the beacons themselves are going to have impact on that. The main study that was done maybe 5 years ago is developing an antibody to a quadruplex, to the specific quadruplex, and that's the only thing that's been definitive. So putting that in, and if it binds, then the quadruplex is there. So yeah, it's very difficult to say that it is structurally formed within inside the cell, but we are relying then on the phenotypic change.
Yeah, I was just wondering if the changes you saw in your assay would be due to the kind of physiological conditions, and then further on, what's regulating inside the cell, the machinery that's regulating the quadruplex structure.
Yeah. Yeah, it's quite difficult to say at this point. I think the evidence is building that the quadruplexes do play a very important regulatory role. So for this research, it's very much assuming that to be true and going through then the library of the structures that we're finding and seeing what the phenotypic changes are, so that we can build that library. And then once we see something that has merit, then we will need to go back and prove that is actually what's happening.
How do these get into the cell?
At the minute they're just getting in themselves, because they go quite globular. They're very easily
actively taken.
Actively uptake, yeah. But towards application, I'm envisioning a phagosome or liposome, some sort of packaging to specifically target
delivery vehicle.
Yeah.
Hello again. So it's Vas Bavro from University of Essex. So Essex is also quite teaching intense, but we are trying to change that. Maybe you've heard that we've been the University of the Year last year, and we are going up and up and up. So hopefully that selection of slides will convince you. Although I haven't structured it exactly in the sense of bottlenecks, etc. I'm grateful to the previous speakers that they've introduced the topic of the Efflux-Pumps, because obviously what I would like to say, I'm actually not per se a compound person. I'm coming from a structural perspective, but what good is a compound, and John mentioned that before, if it couldn't get into the cell? And even if it gets into the cell, there are these systems that will intercept it and throw it out. So we need to address this vast gap of understanding of how these efflux systems work. Can we actually, at some point, get effective efflux inhibitors? And what I wanted to see as a bigger picture, and that's what my second part of the talk is, although it is not connected directly, but it's more of a integrative part of it. We would like to understand the integrity of the cell envelope and how things come together and why some things could go in and why some other things could not come in. And that is, I think, a bigger picture that is often lost behind these trees of trying to find the super effective compound. Okay, so my mouthful of a talk is going to be rather simpler. I'm going to break it into two parts. The first part is actually kind of a, well, my toy project almost for forever. I did my first postdoc in Cambridge in the lab of Ben Rizzi, when we started looking at TOC. TOC is quite a famous molecule. It looks like a tube, almost like a telescope. It has an aperture on its end, and there's the periplasmic end. So what you're looking here is the double membrane of gram-negative bacterium, the ones that we mentioned before that are difficult to treat. And TOC sits on the outer membrane, and then different parts, which we call R&D Efflux-Pumps, assemble to the bottom of TOC, creating a channel that spans presumably either the periplasmic space out, or even, there are some suggestions, from the cytoplasm out. So what is that machinery doing then? It is actually actively pumping antibiotics out of the cells, lowering the active concentration within the cell. So to understand how all this works, as a postdoc actually solved the open structure of this molecule by crystallography. And this is a membrane protein, and crystallography of membrane proteins is notoriously difficult. Took me some 4 years to do that, and well, in the meantime, we also have great advantages of cryo-EM being developed to a point when we could actually see molecular detail. And I would like to say that this is probably one of the bottlenecks that could be discussed from a structural point of view, is if we want to understand membrane proteins. I think now finally we are getting over this first bottleneck, the inability of getting good quality crystals. And the advent of cryo-EM is fantastic. A bottleneck is that these machines, the cryosys now, are quite expensive and not available to everyone, not to people like, I don't know how it's Anglo-Ruskin, but definitely not like Essex. So we're looking for collaborations far and wide so we could work on it. And talking about membrane protein crystallography or structure biology of membrane proteins, this a major problem related to Efflux-Pumps. And now again, we're talking about one particular family of these Efflux-Pumps, but it's actually quite a widespread issue. So Efflux-Pumps have mostly developed in commensal bacteria to deal, in enterobacteria in particular, to deal with the actual environment to which they're exposed in the gut of the host. And the gut creates bile acids, which are actually effective detergents. So many of these pumps have evolved very good strategies to recognize detergent-like molecules and pump them out. This is a big issue in when you try to actually study these pumps in crystallographic setting, because if you want to put a molecule of interest, and in this case we're of course talking about antimicrobial compounds, we're interested how the drug binding pocket of an efflux pump recognizes the antibiotic of interest, for example for a quinolone, and pumps it out. However, every time you try to crystallize something in the presence of detergent, and detergent is needed because you need to extract the protein from the membrane, you saturate the sample with detergent, which being a substrate substitutes the molecule of interest. You end up time after time with the same protein with a lot of detergents in. So one of the high resolution molecules here, this is an experimental crystal structure. You could see the detergent molecules bound, including several of them, in the drug binding pocket of ACRP, one of the prototypical R&D pumps, which I'll be talking about. So at one point, I was still in Birmingham, by the way. That was when I was a lecturer / a Birmingham fellow, and I had this idea, looking into this problem and realizing that we have detergent bound within the molecule, that we need to circumvent it somehow. And in particular, I was interested in this mutation in Salmonella pump. At the time was not available, the structure of the Salmonella ACRB, which actually this mutation in the drug binding pocket converts it to a very effective pump for fluoroquinolones in particular. So it becomes pretty much resistant to fluoroquinolones, and the previous talk actually mentioned that fluoroquinolones target the gyrates. But a particular mutation in the R&D pump could also be stimulated by exposure to fluoroquinolones, and it was the first time it was actually described in the group of Laura Piddock, with whom we collaborated. So we looked at how could we actually go around and figure out what's going on in the pocket. So the suggestion how to circumvent it is actually not just smart, but small. I guess you might have heard about the SMA technology. Who has heard about it? Great, so many people. That I do not need to introduce it in very many details, but unlike the detergent, the benefit of styrene maleic acid copolymer, or SMA, is that it's, first of all, it's a very large polymer, and by its very chemical nature, it is not a substrate of the pump. So specifically for R&D transporters, which are the major efflux pumps in gram-negative bacteria, this does the trick. Essentially, we could apply a pH-sensitive extraction of the membrane using SMA, and what it does this kind of cookie-cutter scenario when it actually engulfs the native membrane around the transporter. And why is that important as well as everything else? The native membrane often has a lot of allosteric effects that were overlooked as well. So I placed that at the time for a Wellcome Trust SID award, which we successfully got, and in collaboration with University of Leeds, we actually, these two guys, Steve Munch and Vincent Postis, we got a structure of the first cryo-EM structure, I should say, of Salmonella ACRB. I mean, this is not published yet, so this is one of the first times I'm showing it, the 4.1 angstroms thereof, and we actually get the beta data set now, and one now with erythromycin in it. So basically the point I'm trying to say, we have a technology which we have developed and we think could address in detail this deficiency of detergent substitution within R&D pumps. Whether it is extensible to other types of transporters, I believe yes. Whether it is extensible to other types of membrane proteins, you already know yes. So we are probably looking for collaborations to exploit that and maybe bring it forward for a bigger award, and we would need more work at that. And again, something that John said, if you have a good understanding of the target binding, and even if it's a lead compound, what you could do, and this here are some lead compounds, that are not our work directly, but our collaborators' work, and you could see that this is an initial compound that has been co-crystallized in a different way with a R&D transporter. And then if you look into the crystallization conditions, you could see how you could actually extend the compound further, and this is two-way iterative bioinformatic work. You could grow the compound by analyzing exact modes of binding and exact crevices where you could expand. So this kind of back and forth evolution of compounds for in silico screening combined with high throughput crystallography, and I want to say that at Essex actually we have quite a good crystallography division. And one of my colleagues, Filip Oprisky, he's actually developing a high throughput crystallization platform that would allow you to screen multiple target compounds in that very fashion. So that's in crystal screening for compounds for binding. So this is something that is also quite an interesting perspective. And now, as I said, the Efflux-Pumps obviously are one of the major contributors for the resistance, but not the only one. I mean, when we are standing from this viewpoint of what does the envelope of the cell of the gram-negatives in particular do, it not only do they efflux things out, but they actively prevent things from getting in. And there are many sort of gateways into the cell, but probably the one that is critical for antibiotics, for the size of the antibiotics that we're interested in, which are effective against gram-positive bacteria, these are the porins, and especially trimeric porins, like ONPEF, they're ubiquitous. I think you probably know about them. These are big beta proteins that allow compounds for up to maybe 1,000, less than that, but about 800 Daltons to pass through. However, we often misunderstand the complexity that is provided by LPS, for example, which is a critical component of the gram-negative envelope. So there is a project that we started while I was in Birmingham with Adam Cunningham, who was really interested to understand how porins and LPS could be exploited for making of vaccine towards Salmonella. Again, Salmonella is one of my pet projects. So one thing that he was wondering was why is that only some antibodies are protective. When you get Salmonella infection, you get a lot of antibodies which target surface epitopes, but only some of them are really protective. So we try to understand what's going on. This interplay is organismal level, is at the level of the IgG, obviously, but also we need to understand what the bacteria is doing. So that is where this antibody-antigen interactions are important to be seen in the actual context of the cell envelope. So that's Adam. You might have seen him if you landed on the airport in Birmingham. He's one of the heroes of Birmingham. Okay, this is a very complicated slide, but it's not really. So we have Salmonella typhimurium and Salmonella enteritidis, wild type, and we have found that we could get a very good antibody based on, I'll just take 2 minutes again, OMP-D, a trimeric porin in the case of Salmonella typhimurium. Now, if LPS is present on the surface of the cell, although it's going to kill the Salmonella typhimurium, it doesn't quite kill enteritidis. But on a naked or rough strain of Salmonella, the killing is present for both. So this could be summarized like this. We looked at that and it looked like, okay, if we have an antibody that binds to this protein, is it as simple as that if we have an occlusion of sorts that hides the epitope, yes or no? Just to point out that this really is dependent on IgG, and if you knock out particularly IgG1 subtype, you get a big bacterial load. So what is happening is perhaps too difficult to see yet, but an aha moment was when I was talking with one of my collaborators who actually does full atomic simulations of bacterial envelopes, and that's J.C. Gumbert at Georgia Tech in the States. So J.C. have created a very explicit model of LPS, which kind of gave us an appreciation of how dense actually this network is. And I asked him, could you put a trimeric porin there just for a laugh, let's see. So he did that. I looked at some antibody structures and very plainly just tried to dock one in. And I realized that you could only about dock one FAB fragment per a footprint created by a trimeric porin. We actually could do that in extended molecular dynamics, and to see that an antibody would indeed be able to just about access an epitope at the surface, and this is the actual one single amino acid epitope that is differing between the two OMPs in this case, and binds through this mesh of very dense LPS. That is only possible for trimeric porins, and by the same prediction, an antibody would not be able to access a monomeric small porin such as OMP. I'm going to keep this short, but actually that's what we could prove, and hopefully the paper showing it is going to come through very soon to give you an idea of this very spherical and dynamic interaction on the surface. So we have a lot of interactions with Birmingham still and Georgia Tech, and I would like to also thank particularly Coral Dominguez Medina, who did a lot of this work in Salmonella. So thank you for your patience.
One quick question. Thank you.
The antibodies obviously they can't get into the outer membrane, the protrusions. But
it's more complex than that, because in order for a good opsonization to happen, both sort of FAB fragments have to be engaged with the antibody, with the antigen. There is a particular density of antigens that has to be reached for an efficient killing complex to be created and the complement to be activated. So there's a lot to be said about it. I'm not that much an immunologist to be able to answer that in particular, but yes.
Because always the LP is actually stopping. Okay, can we use the same approach? If you try in a cytoplasmic membrane, other transporter like SMO or MAD transporter, can we use this approach? If the antibodies are small enough to...
This is not against the transporter, this is against the non. So transporters and the inner membrane, they're not...
I know, we are, the one you're targeting, only the YMP.
Okay, yes, that's correct.
So can we use the same one? Because if you can't access from the LPS layer, and why can't you try in a different approach to...
I'm not sure I get the question.
I'll talk
to me later, because what we're trying to show here, there's this a big barrier to antibiotic actually entry as well, which is provided by LPS and the pods. That's the only thing that's related really to this talk. I just wanted to bring this in to provide a bigger view, bigger sort of perspective on the complexities of the envelope, which are often lost when we're looking at particular interactions just at the level of transporters and so without understanding what's happening in the outer membrane, I think we do not have a good compound, because a lot of this accessibility issue that the antibody is facing would be faced by large antibiotics. So the same thing that we might be targeting maybe particular OMPs as well to try and leave them open for longer, getting compounds that could possibly do that.
Absolutely.
It's something, that is obviously open to discussion.
All right, afternoon everyone. So I'm a senior lecturer in microbial biochemistry, School of Biosciences, University of Kent. So over the last few years, my group has become interested in understanding mechanisms of antibiotic toxicity. Not really primary modes of action, more secondary toxicity related to bacterial respiration. So I'll touch on this very briefly soon. I haven't really got time to go into it in much detail. Also, really just started working on antibiotic discovery as well, so we're quite new to this field, but hoping to move into this area a little more in the coming years. So, this is basically what my lab does. So, part of what I do, I work on bacterial biofuels. I'm not going to talk about it, it's not very relevant to today's session, but the bulk of what we do is we're interested in stress responses in bacteria. So like stresses encountered during infection and how the bacteria respond to them. So we work with gram-negative bacteria, actually E. coli mainly. We've got a lot of clinical isolates that we work with. And I'll give you an example. We're interested in how they respond to nitric oxide. I don't know where that's gone. So I made this on a PC, so it's punishing me now. So, how this links into bacterial respiration. So, why is this relevant to this session today? So, actually, bacterial respiration and antibiotic resistance and antimicrobial efficacy. These two are actually quite closely linked. Okay, so I'll touch on this shortly. So this might be of interest to some of you. All right. Okay. So some projects that we're running at the moment. So what do? So we work with a lot of coli clinical isolates. So we've got quite close links with local NHS, and we collect these isolates, sequence the genomes through collaboration with Ian Goodhead at the University of Salford. And we'll mine these genomes. We'll take a look. We'll predict resistance profiles, and we'll do the susceptibility testing, that sort of thing, and generate like suites of isolates that some of you might be interested in. One of the main interests is this link between respiration and antibiotic susceptibility. Okay, so for certain classes of antibiotic, if you shut down respiration, you get a huge change in antibiotic susceptibility. So this is one particular data set. So if you shut down respiration, you get a large increase in tolerance to certain classes of antibiotic. Different classes of antibiotic, you add respiratory inhibitors, you actually get the opposite effect. So it depends on the antibiotic that you're using. So it's not really related to the primary target. It's more like secondary toxicity, so like generation of reactive oxygen species, alterations in the proton motive force, which can impact upon uptake of antimicrobials. I suppose the Efflux-Pumps as well is important. So it's quite complex, and we're kind of interested in teasing out the underpinning mechanism why respiration impacts upon antimicrobial efficacy. All right. Okay. So we've been involved in antibiotics on Earth for the past few years. So we work with the schools and get various environmental isolates, and we'll do the screening with them, and we sequence the genomes and take a look and see what they've got basically, and try and work out using some in silico approaches, what antibiotics they might be producing. But to be honest, I'm not that good at that. It'd be nice to talk to some of you that have a bit more expertise in that area. A brand new project that I'm pretty excited about. So I've just started a collaboration with a group at the University of Ghana, and they work with these endophytic fungal isolates. So these grow in a marine environment. So this might be of interest to some of you. So associated with seaweed, and they do a lot of the fractionation of the extracts that you get from these fungal isolates. And they're interested in testing a range of microbial pathogens. So what we'll be doing is screening sort of bacterial isolates in the near future. Also at Kent, there's quite a lot of work on infectious diseases. So some of my colleagues work on fungal pathogens, Candida, Cryptococcus, also eukaryotic parasites, Blastocystis, Cryptosporidium. There's some virology going on. So we've got quite a tight network of PIs at Kent that are interested in looking at I suppose testing novel antimicrobials, the efficacy with a range of different pathogens. Right. Okay. What techniques do we use? So do a lot of bacterial culture, I suppose. I have a history of doing a lot of fermentation. We do a lot of anaerobic culture as well. Physiological measurements, respiration rates, that sort of thing, we're pretty good at. We genome editing techniques using lambda-red recombinering techniques in coli, for example. Sequencing in collaboration with Salford, so we've done a lot of the sequencing ourselves and the genome annotation. Something that we're just getting into and kind of developing that skills base in the lab, which is quite fun and exciting. Been having to play around with MinION technology recently, so that's something that we're a number of PIs are kind of quite interested in developing. Analytical tools at Kent. We've got a lot of good analytical tools. So we've got UPLC QTOF Waters instrument. We use that quite a lot. Moldy GCMS. We've got a 600 NMR machine. We're quite well set up in the department for analysis of small molecules. Not that I do a huge amount of that, but it's there's expertise on site, which is really quite helpful. Really, once we start to get some really promising hits. Drug screening. So so we tend to do this the hard way with kind of BSAC methods, viability assays, that sort of thing. There is a robot in the department as well. So when we get access to different libraries of drugs. Also, I'm program director for the MSc Infectious Diseases, so we have an annual army of students, and we run projects. And this cohort of students, they like to do kind of drug screening assays, that sort of thing. So we have some kind of manpower for kind of smaller projects, 3-meter projects, things like that. If you need a pair of hands, if it's quite labour intensive, let's say, the type of work that you want to do, but kind of thing. We've also got the robots as well. So. Yes. Okay. My bottleneck. So I don't really have access to a lot of different natural extracts or drug libraries either. Anyone who's good at bioinformatics, looking at genomes, predicting what these organisms can do, that would be quite helpful. Animal models as well. I suppose we're a little short on that expertise at Kent, but so down the line that will be quite useful. And also structural characterisation, way down the line. It would be interesting talking to anyone who has expertise in that area. I've stuck to 5 slides, as I should do, and so I'm not able to thank anybody. But I do actually work with a lot of very, very helpful collaborators at Kent. But okay, thank you.
Any questions for Mark? So what kind of bioinformatic tools do you use at the moment to identify pathways?
Well, just play around with anti-smash basically, but I don't use it much. It's like, I'm not very good at that sort of thing. But I haven't got time. So any other suggestions? Anything else that's working well for you guys? It'd be good to kind of have a chat. And I'm sure there's plenty out there. I just don't know a lot about it.
Just to follow up to that, I'd agree that anti-smash is one of the ones that we do use, and there are limitations of any of these tools that rely on kind of things that are similar to things we already know about. But I was talking to somebody in Nathaniel Martin's group in Leiden a couple of weeks ago, and they're developing some new tools. So I think it's an area that we will see new tools coming soon, which will be good.
Yeah, that will be helpful.
Lovely.
Is that it? Thank you.
Thank you.
Okay. Good afternoon. My name is Soumya Khalil. I'm from the University of Aberdeen. I might be the furthest traveled yet. Right. So within the university, I run something called the Scottish Biologics facility, and we work with large molecules. So we're slightly moving away from the world of small molecules into antibodies. That's my background is in antibody engineering, and within the facility, I run a couple of projects developing novel therapeutics and also diagnostics for the detection, diagnosis, and treatment of bacterial and fungal infections. So I'm going to talk to you about those projects and also what we do in the facility and how we can contribute towards this network. So the Scottish Biologics facility, as I said, is a biologics drug discovery centre. We are based in the University of Aberdeen, and we work with both academic groups and commercial organisations. We specialize in making monoclonal antibodies. These are recombinant antibodies and also peptides, and against a range of targets. And these targets could be proteins, peptides, or small molecular weight compounds, which are otherwise known as haptens. These antibodies can be developed into reagent tools or diagnostics and therapeutics. Right now, there are 4 drug discovery projects in the facility which I run, and this is anti-infectives. We have antibacterial and antifungal programs. There's one in neurodegenerative diseases. This is mainly focused on Alzheimer's disease, and there's also programs in inflammatory liver disease like the NASH. So the technology that we use is phage display and binding site reformatting. So phage display is basically working with large antibody libraries, and we have libraries which are either naive, which is isolated from human blood, or we can also prime an animal like mouse or sheep with the antigen of choice, and we can make antibody libraries, and we can fish out the antibody binders that we are interested in, and we use phage display for that fishing out. We can also reformat the binding sites. So in phage display, we work with antibody fragments. We are not working with full IgGs at the hit identification stage. later on we can reformat that into soluble antibody fragments, bispecifics, or full IgGs. So we'll talk a little bit about today's theme, which is antimicrobial resistance. And we all agree that it is one of the major challenges faced in anti-infectious drug discovery. Usually we get treatment which is based on broad spectrum antibiotics, and this is not helping the case. We should be looking at more treatment strategies which are pathogen specific. And in the world of drug discovery, antibodies and specificity goes hand in hand. So we look at monoclonal antibodies, which can be developed into antibacterial or antifungal therapeutics. The other advantage of using antibodies is that we can harness the power of the immune system. So we can recruit components of the immune system which can come and clear the infection. So far there are 5 biologics drugs approved by FDA as antimicrobial agents. And if you look at look into the clinical trials, there are a lot of compounds. These are mainly the antibodies, human or humanized MABs that are developed against gram-negative and gram-positive bacteria right now. So in the light of AMR, I think we need to think slightly different. We need to develop novel drug discovery approaches, and this is mainly targeting proteins, which are essential for the viability of the pathogen during infection. We should also look at bacterial virulence pathway rather than just developing drugs which inhibit growth or kill the bacteria. We need to move away from this broad spectrum towards narrow spectrum disease-specific drugs, and also alternative therapies like peptides, antimicrobial peptides, monoclonal antibodies, or phage therapy, and the immunostimulatory products such as CAR-T, which is quite upcoming right now. So I think we need to start looking into these kind of approaches. And this is not going to be a reality unless we have improved diagnostics. So also we need to invest a little bit more into the diagnostic area as well, develop point-of-care diagnostics, which can differentiate between bacterial and fungal infections. Also, narrow spectrum diagnostics for targeted therapy. This is, for example, in the case of cystic fibrosis, you get infections which could be Pseudomonas or Burkholderia. So if we have diagnostic tests which can pinpoint what type of infection is that, then we can just treat that particular bacteria. Also tests to detect resistance so that clinicians can prescribe drugs based on the drug resistance of the bacteria which is or the pathogen involved. And we have to move slightly towards personalized medicine, like what happens in cancer and autoimmune disorders. Use the power of monoclonal antibodies and immune system and just stratify patients and develop treatments accordingly. So this is one of the projects that we have in the SPF. This is against gram-negative pathogen, which is Pseudomonas. We all know it's a versatile pathogen, mainly because it develops resistance at a rapid rate, and also it expresses a lot of virulence factors. And in most cases, these virulence factors are the actual causes. It actually causes damage in the host tissue rather than the bacteria itself. And in Pseudomonas, these virulence factors are under control of quorum sensing or cell-to-cell communication. So our strategy was to develop monoclonal antibodies which can break that quorum sensing or cell-to-cell communication so that we can reduce the expression of virulence factors. And because these antibodies are binding to the cell signaling molecules of Pseudomonas, which is actually on the outside of the cell, there is less chance of resistance development using this technology or this strategy. And by recruiting antibodies to the site of infection, we can enhance the clearance of bacteria by other components of the immune system. So what we have done is, in this particular project, we immunized sheep with homoserine lactones, which is one of the prime quorum sensing compounds in Pseudomonas. After sheep immunization, we collect sheep blood after a few rounds of immunization and construct an immunized sheep library, an antibody phage display library. And using phage display technology, we fish out the antibody binders. And these antibodies that we isolated for this particular project, I call them HSL2, HSL4 because they are specific against homoserine lactones, the particular class of homoserine lactones in Pseudomonas aeruginosa. We tested them in a non-neutropenic lung infection model, and these antibodies actually give a protection. It improves the survival of mice in a lung infection model. So it's actually giving protection in a Pseudomonas infection. So that's quite encouraging. And we also thought about developing a diagnostic based on these antibodies. Because again, these antibodies are quite specific, and it also has nanomolar sensitivity. And it's quite challenging to make antibodies to small molecules, and with that level of sensitivity. With sheep immunization, we managed to do that. So we thought, why don't we develop diagnostics specifically looking at this biomarker homoserine lactone in cystic fibrosis patients. So what we wanted to show that in cystic fibrosis patients, there are elevated levels of homoserine lactones in sputum, plasma, and urine. So we need to show that these antibodies work in complex matrices such as urine. So we grew Pseudomonas in human urine, and Pseudomonas secretes homoserine lactones. So this is a competition ELISA format, but what it's showing is with these antibodies we can detect the HSL molecule secreted into the culture medium by Pseudomonas. And it's not losing its sensitivity by too much. So in PBS, the detection limit is 1.5 nanomolar, whereas in urine it's still in single nanomolar IC20 value. So we're collaborating with University of Edinburgh, Professor Till Bachman, and we're developing a diagnostic test system. So these antibodies will form part of a test system which can detect biomarkers in sputum or urine samples in cystic fibrosis patients. And this can be a point of care diagnostic test kit. So what they've done is they've immobilized HSL antibodies onto the electrode. This is chemisorption, and it's a mock wound fluid assay that they've set up. So they spiked wound fluid with homoserine lactones, and they're getting a detection limit of 1.7 picomole per milliliter, which is quite within the range of what is seen in cystic fibrosis patients, sputum and urine. So we are in that, we are right in that limit of detection range, which is quite encouraging. So the next stage will be using real patient samples and seeing whether this can be, this can develop valid data. So the second project we have is an antifungal, and again focusing on Candida albicans for this particular project. Just a bit of stats, invasive fungal infection causes over 2 million infections in immunocompromised patients, and the death rate is quite high in this in the case of fungal infections, up to 50%. Antifungal therapeutics pipeline is quite sparse, and we need novel therapeutics, just like what we need in antibacterial. We need more therapeutics in antifungal area. And also there are drug-resistant species reported like Candida auris. So this project is in collaboration with the Aberdeen Fungal Group, and so what we have done is isolate. So we have isolated anti-Candida antibodies which target the cell wall proteins, and we have shown here that these antibodies can reduce fungal burden in the kidneys of in a mice model of infection. So if you look at this group 1 here, this is antibodies which is administered before as a prophylactic. So here we have shown that these antibodies can work very well as a prophylactic agent for people who are suspected to have fungal infections, like in immunocompromised patients. So last slide, just quickly, what we can provide. You've heard we have strong expertise in developing recombinant antibodies. It can be fragments or full IgGs. And it can be high affinity and epitope specificity. We can co-develop pre-screening experiments. We work with Galeria. A few people mentioned Galeria here. Also with C. elegans, which is the nematode worms. We have a lot of expertise in developing therapeutics in vivo diagnostics, and also commercialization of research assets from the university. So we have spun out companies from the University of Aberdeen. The challenges are limited resources to identify novel antimicrobial targets. So we'd like to talk to people who have new ideas, new targets, and if they are looking for antibodies to bind to their targets, we are the right choice. And also, it would be nice to develop some high throughput assays for the identification of these biologic binders. We are also developing to collaborate with groups who are looking at alternative therapies, who have expertise in bacterial and fungal pathogenicity, and we're quite keen to develop proof of concept experiments. Thank you.
We've probably got time for one quick question. Anyone's? Yes, sir.
So the Candida auris is the newly emerging, antimicrobial resistant Candida species.
Yeah.
So in terms of diagnosis, how do you distinguish between Candida albicans and Candida auris?
So it has to be, so we are working with a range of cell wall proteins. So there are cell wall proteins which are specific to Candida auris. So the animal model that I showed is for Candida, specific for Candida albicans, but then we have antibodies in the pipeline which are specific against Candida auris. So they could be used for Candida auris detection.
Sorry, very quickly. Do you do nanobodies as well?
Nanobodies,
Yeah.
we have, yes, there is a company spun out from the university which works with shark antibodies. We don't do the camel ones,
Right.
but we can do single domain binders,
Okay.
yeah.
So, as a service or as a collaboration?
Both,
Okay.
both, yeah.
Yeah, we'll talk now.
Yeah.
We'll speak later.
Yeah,
Excellent.
Hey, afternoon everyone. Thanks to the organisers for giving me a chance to speak today. I'm going to change tack slightly because I don't actually currently work on discovering new antibiotics or antimicrobials. I'm a microbiologist and I study evolution of resistance. I'm an Industrial Innovation Fellow at the minute, so I've been working on this assay which can be used to for environmental surveillance of antibiotics and determine the concentrations which select for resistance. And the reason that I asked to come and speak to you all is that I'm hoping to test some of the compounds that you may be developing, or perhaps compounds which you started working on and had to abandon for whatever reason, as sort of a proof of concept that perhaps the assay that I'm working on could be useful in antibiotic development. So we all know about the broken pipeline. The angle that I'm coming from is that we're trying to protect and do good antibiotic stewardship of the antibiotics that we already have. And while most of you are all really struggling to develop new antibiotics and get them through the pipeline, that should make it even more important that we try and think forward to, once you do get things through, what can we do to try and stop the emergence of resistance. So the reason, or one of the reasons that the antibiotic development pipeline is broken is that antibiotics are high risk and low profit. So they take years to develop, they're only for acute illnesses, so they have low return. These first 2 points, as a microbiologist, I feel are beyond the reach of what we could try and impact. They're more financial problems, whereas the last point is they're risky to develop because they soon become ineffective due to novel resistances arising. So what I'm proposing is that perhaps we could take the approach of green pharmacy when developing novel antibiotics. And there's a sort of paraphrase definition of green pharmacy in the corner there. And basically it's about trying to reduce or minimize the entry of active pharmaceuticals into the environment, but also to improve their efficiency and effectiveness in healthcare. So the assay is called the Selective Assay for Growth-Based Endpoints. I left a lot of time because this is sort of a new idea and I wanted to get people's opinions on what they thought of this, if you had any knee-jerk reactions. So yeah, that'd be good. Thanks.
We've got plenty of time for questions. Any burning questions? Yes.
This is more of a comment than a question. So I wrote a review with Laura Piddock on abandoned antibiotics, which has got a database called Antibiotic DB. Anybody can access and find antibiotics that were in development, have now been abandoned, the reasons and whether they're worth taking. It was published in JAC last year, I think. The Antibiotic DB is just search for that and you'll find it.
Great, thank you.
What strains are you using in your assay?
So the assay is actually based on a complex community of bacteria. So I use untreated wastewater, which would include all of your, it's supposed to be sort of representative of the human gut microbiome, but of course, you'd have shedding from showers and stuff all goes into wastewater. So it's sort of, you screen against everything and then you can go in and see what individual strains is being enriched, yeah.
Okay.
Do you have any gram positives within that assay? Because if it's sort of representative of the gut microbiome, you're less likely to have gram positive organisms, one assumes.
There are a few gram positives, but it's mostly gram negative, yeah.
Which sample from the wastewater treatment are you using? The one that is activated or the one after the first filtering step?
So it's the stuff that goes in after the first filtering step, so it's a liquid
isolated one
Yeah.
outside now.
No.
Just wondering, are you testing some of these drugs in combination? I think we've got a mycobacterial expert up there. One of the problems necessarily that things done is this push to have single drug therapies for treatments, which spontaneous resistance rates goes up and things, and you see a big difference when you use drugs in combination. So perhaps some of these drugs that are coming online with these because of the sort of commercial pressure to push single drugs rather than treatments that work based on multiple drugs. And maybe one of the ways that you could show its activity is to show that combination of those drugs lower resistance.
Yeah, so we can use the assay to look at more than one compound at the same time, so mixtures, and we are doing that currently. The ones that we've been looking at the moment, worryingly, they suggest that when they're used in combination, it actually lowers the concentration at which you observe selection for resistance, and that's a commonly used combination, so that's kind of concerning, actually.
Yeah, so I think it's really nice that you use the whole community to sort of worry when you use antibiotics, lots of microbes in there, you can be affecting something completely different. So yeah, like I say, when we're using these novel antibiotics, there might be creating resistance towards other compounds. And again, with this dual therapy combination idea, what exactly, other than developing this lower threshold, what resistances are occurring in that community? I would imagine the more antibiotics you use, the more problems you're going to get. Because as you kill him, I think, I would say, you're going to cause more problems.
Yeah, I agree. I think that's just the way bacteria are going to evolve resistance.
That's great because you can address that.
Yeah, so we do metagenomics as well on the evolved bacterial populations, and we can observe co-selection, which is what you're referring to, I think. So we expose to cefotaxime, for example, and we see aminoglycoside and vancomycin resistance genes go up, for example. So yeah, we can look at that as well.
Have you benchmarked your assay system against other assay platforms that can also make predictions of or likelihood of resistance? So you're thinking about systems like the Topoisomerases platform and Discover system. Have you looked at how your data compares and what you think in that system?
I haven't heard of that, so no, but I'll try and find that later, or maybe get it from you again, because I'm not going to remember the name.
It sounds like it's quite similar to what you're trying to do.
Yeah, so this is actually a really quick assay, so you only run it for 12 hours and you get the results really quickly. It's actually based on more long-term evolution experiments, so evolving the community for 7 days and then doing qPCR and metagenomics. And based on qPCR data, the endpoints that you get with this much quicker method, they're very similar in terms of what the lowest concentration that selects resistance. So it's been verified against qPCR data, you see, and that's for eight antibiotics across 6 different classes that I've tested so far.
The last question. Thank you.
I'm looking at bioprospecting and I'm trying to add into antibiotics to see if they can lower the concentration of antibiotic required. Would this work for my bioprospecting if I don't know any genes involved and they are killing the bacteria that I'm testing? Would you be able to just, would this work for a compound that hasn't been done before?
That's exactly what I want to do. So yeah, we can have a chat and yeah, that would be good.
Matt promised this is really fast.
Just a comment, really, a knee-jerk reaction and feedback. I think is a great idea and a great approach. But what I do like as well is the fact that you are just sort of throwing this out there. Let's sort of work together with this new approach. So it's again speaks directly to ABX's initial idea. So thank you.
It's fantastic. Excellent. Thank you very much.
Thank you.
So I am going to talk you through my PhD project. I'm a first year PhD student in the niche microbiology group in Ulster University in Northern Ireland, so I apologise if I have to listen to the Northern Irish accent for the second time today. But yeah, so my project involves pheromone-based communication within enterococci. So the enterococci are a genus of gram-positive bacteria that are found widely distributed within the natural environment, but they're also found within the gastrointestinal tracts of a number of different animals and also in humans. Within humans, they are present from birth, and the two main species found in humans is Efflux and Efficacy, which you may be familiar with. These can be isolated from the oral cavity, but they are also found mainly in the gastrointestinal tract, specifically in the colon. Their numbers are at their highest. As with the number of bacteria, if you give them the opportunity, they will act to cause infections. The most common ones they cause are actually hospital-derived infections like UTIs, bacteremia, and then also endocarditis. These are all more commonly caused by Efflux, but infection with Efficacy is associated with greater antibiotic resistance. So a number of different cells, or nearly all kinds of cells, take part in some sort of cell-to-cell communication. This basically consists of them utilising a signalling molecule that accumulates in their environment as the cell density increases. And this allows them to obviously monitor the cell composition and also the cell numbers, and then alter their gene expression according to the information they receive. So I'm sure you're all aware that gram-negative cells tend to use homoserine lactones, whereas gram-positive cells tend to use peptides for cell-to-cell communication. In my project, I am looking at pheromone-responsive plasmids, which use a peptide signal to initiate the transfer of plasmid DNA between cells. This is the system for the PCF10 pheromone-responsive plasmid system, which you can see is just represented in a donor cell. This plasmid encodes tetracycline resistance, but this plasmid is one of over 30 that have been identified within Efflux and Efficacy. This system basically consists of the recipient cell, or the plasmid-free cell. It's the one that produces the peptide signal or the pheromone, and it's excreted into their extracellular environment. It's then recognized on the donor cell by a plasmid-encoded receptor, PRGZ, and it's then actively taken up inside the donor cell, and this is an ATP-driven mechanism. Once the donor cell has taken up sufficient pheromone, it then transcribes a number of genes from PCF10. This results in the expression of a number of proteins. One of these is the surface protein, ASC10, which is an aggregation substance that allows the donors and the recipients to actually clump together. And a second is a mating channel, which is a type 4 secretion system homologue, and this allows the donor to pass the recipient then a copy of the plasmid. So my project is exploiting this bacterial cell-to-cell communication system for delivery of substances to at least the bacterial cell wall. So a number of bottlenecks in my project include, firstly, the pheromones are very hydrophobic. This is the structure for CCF10, which is the pheromone that induces the transfer of PCF10 plasmid. It's very hydrophobic, and that obviously doesn't mix well with the aqueous environment that I grow my bacteria in. And secondly, they are peptides. So in theory, they are susceptible to proteolytic degradation and also just general degradation when in solution. And then if we were wanting to utilize this later on in my project within animal models or something similar, it's likely that they'd be cleared through the kidneys very rapidly because they're only 7 amino acids long. And I am probably going to have to protect them to overcome these issues. So using something like pegylation, but in doing so I may then sterically inhibit their function because they're so small. So I'm going to have to find some sort of in-between to allow them to still work, but also at the same time protect them. So thank you for listening. That was very quick. And thanks to my team back at Ulster and to the guys who are organising it. Thank you.
Thank you. Any questions for Amy? Yes.
Do you all know peptides, or are you also working on trying to find new ones in the other regard?
All of the plasmids, the pheromone-responsive plasmids that are known, only about 25 out of the 30 have actually had their pheromones defined. 5 of the plasmids are best defined, and everyone seems to care more about 5 rather than the rest of them. And there's also some that no one's even bothered looking in other species. So the 5 that have been mainly looked at is the ones I'm looking at, just because it's easier for me, which is not obviously ideal with the issues that we're dealing with antibiotic resistance. But I will be looking at the main 5 pheromone-responsive plasmids and their pheromones.
Sorry, what you refer to as receptor, is it an ABC transporter actually?
No, it's the receptor. They utilise the system in, I highlighted, the OPPS is the chromosomally encoded uptake system. Okay. So all the cells should in theory have that, and it's just the PRGZ that is the receptor.
Okay.
Are there any novel sort of methods that you can use beyond sort of just protecting your peptide for delivery to sort of infection site? Is there
I've
anything?
thought of like liquidation or some sort of protective nanomolecules or something. I'm not entirely sure. That'll be a bit later in my project, and I haven't really given it much thought at this moment.
Or some means by which to actually express that target, express that peptide at the site, rather than trying to get peptide to site, perhaps.
Yeah.
So, yes, sir.
Have you thought about just making a, because they're pretty modest in size, why don't you just synthesize a peptoid that's got the same side chain configuration that doesn't have the peptide bonds in it, and then you get around the issue of proteolytic cleavage and
Thing is, because they're so small, they change in amino acids can mean that they just don't work now. They're just not recognised.
You can retain the side chain conformation with the peptide bond. So that would be recognised in cleave, but it's still probably engage the target.
Okay, I'll definitely have to look into that. Thank you.
If you know if it's synthetic chemistry,
yeah.
probably look that up being pretty sure.
Okay.
That hydrophobic should not be easily cleavable, by the way. Most of proteases will cleave at hydrophilic sites.
That's what we're thinking, it's probably a protective mechanism. That's the reason why they're hydrophobic.
Any other questions? No? Thank you very much, Amy.
Okay, good afternoon everyone. So I'm a new lecturer at the University of Kent. My background is very much in enzymology, in vitro systems, and studying natural product biosynthesis. But I would actually say I'm very new to sort of looking at antimicrobials. Okay, so I haven't really discovered an antimicrobial, but I am working on a range of different secondary metabolites from Streptomyces that have potential activity as anti-tumour agents. But that's not really applicable to today's talk. So I'm going to talk about something that may be a little bit random, and that's called cell-free synthetic biology for natural products. So I've worked on cell-free systems. I'm just going to, has anyone ever actually come across a cell-free system, or vaguely as an awareness of what that is? Okay. So cell-free systems, just to briefly sort of summarise this, in vivo systems that make secondary metabolites, as for example, or any given metabolite that you can make in E. coli, Streptomyces, that can take between 1 to 3 weeks or whatever. And I'm particularly interested in actually in vitro systems that either use purified enzymes, cell extracts that contain enzymes that make a metabolic pathway, or even something called cell-free protein synthesis, where you put the DNA in to encode a particular natural product pathway. Particular advantage of this is very, very quick, it's cheap, and also you can look at things that are antimicrobial, which maybe cause problems when you're trying to make that in a live cell. So 2 areas I'm sort of very keen on and very interested in is cell-free protein synthesis, and also another area called one-pot biosynthesis. Now, has anyone seen this figure on this slide here? Excellent, you're not reviewing this. I thought it was highly unlikely. Reviewer number 2 is always the killer, and if you ever had a rejected submission, but this is just a little review. It should be fun. But I'm interested in using one-pot systems where you have purified enzymes or extracts containing enzymes to make specific compounds, and these are a range of different natural products here. One of the ones I worked on here is actually not an antimicrobial, it's a component of vitamin B12. But these are all types of polyketoids, NRPS, PKS hybrids, alkaloids, and these just demonstrate ways of making natural products in a test tube, which actually achieve high yields than used in total synthesis. Okay, just for example. So I'm not going to talk much more on that because I've got students that are going to start working on various projects related to that. What I'm going to talk about is mainly about my experience and what I've published in, and that's cell-free protein synthesis. I'm particularly interested in organisms that make natural products. Okay, so I've worked on a Streptomyces strain called Streptomyces venezuela. I've worked on Bacillus, and essentially you can make an extract from those organisms and study cell-free transcription translation. Now, we've only made, in this example here, simple things like fluorescence proteins, but I've actually started to look at actually making things like peptides and other types of small molecules that are encoded by enzymes. So we can make the enzymes in the cell-free extract and look at enzymatic pathways. An advantage to this, which may have an advantage to antimicrobials, is looking for things that are toxic, which you can't do inside a cell. One of the advantages of cell-free processes is you can automate that and put that onto a robot, so you can do lots of screening assays. And another thing is you can use rapid timescales, low volumes, and these are very cheap reactions. So we can make extracts from different organisms and study protein synthesis essentially. And you're typically looking at about one pence per reaction in a single for a single assay. So I've got a very sort of short. This is only a really sort of short talk here, but I'm working with Christoph Kohr and Emzo from University of Warwick, and we're looking at potential ways of derivatizing, particularly antimicrobial peptides, and trying to put in different amino acids. So there's a paper that will be coming out very soon, not from myself, but there's this interesting study of someone that's showing if you take niacin, the antimicrobial peptide, and take a library of that and look at it in a cell-free system, what they've been able to do is evolve essentially a gram-positive inhibitor directly into a gram-negative inhibitor. And I haven't addressed bottlenecks on there, which I'll come on to in a minute, but one of the major bottlenecks is finding antimicrobials for gram-negative pathogens. Okay, so there's a study that should come out pretty soon showing an application for cell-free in this particular area. I've got experience in using automated systems, robotics for using cell-free systems, and that's what I'm really sort of working on the next phase is using that to study natural product pathways. And one sort of small story that we've been looking at is actually taking biosynthetic gene clusters and putting that into a cell-free reaction. So typically a lot of secondary metabolites you find from Streptomyces are encoded by these so-called clusters, and we can put these into these reactions and get some interesting natural products. And we're at a very primitive stage there, but so it's something we're trying to sort of understand what the caveats of that. There's not really a high throughput study, but we find, for example, if we take some of these operons and split them up into single genes, we actually get a much more productive pathway than working with the native operons there. I don't really read my emails very well, and I actually, it's quite sort of a bit of luck, I actually bought a collared shirt here. And one of the key things I've come across is I didn't actually think about putting a slide about bottlenecks, so I'll do this off the cuff. So I think a lot of people have highlighted some very important points in antimicrobial discovery. I think actually Tony Maxim was highlighting some very important things about trying to bring in people with different skills, different expertise, and not necessarily see an interaction there, but multidisciplinary research is going to be a very important thing. Gram-negative organisms is the hot area, I think. Trying to find antimicrobials. A lot of drugs that we discover in natural products can't get through the cell membrane in a gram-negative. That is one of the key things we've been able to address. I'm not saying my particular technique would be useful for that, but I think I'm just talking out loud here. I think also in terms of bioinformatics, there's a lot of studies, people saying this cluster could make this. I think there's an actual real need to invest more in biochemists and enzymologists to actually work on these pathways. There's a lot of unknown chemistry in particularly biosynthetic gene clusters where there's a lot of hypothetical enzymes that nobody's even touched or worked upon. So I think that's my sort of personal thoughts on that really. So thank you very much for listening.
Gram negs are important, but TV is more important. Any questions? Yeah, Gary.
Just a question about the conversion of gram negative to gram positive or gram positive?
Positive to negative. I think I'm free to say it because the paper's going to be accepted. I reviewed it.
Is it niacin that you use to do that conversion?
Not me. It's another researcher, but they've looked at a library of niacin. They've looked at thousands of different derivatives of the niacin precursor sequence, and they've been involved to see increased activity for gram negs. It doesn't say whether that's been increased diffusion across the membrane. That's not something they've touched upon, but they do show an increased response.
The thing I was curious about was when you see that kind of new specificity, if you like, is it specific to one or two bacteria, or is it really broad spectrum?
So I can't comment too much because it is a paper that's in review. The focus is just a couple of, they've looked at a couple of different types of gram negs, so E. coli. But I I don't want to diverge too much into that.
No, I would expect it to do something like that. You might want to do more work specifically.
So the advantage of this particular technology and method is you can make antimicrobial peptides in a very small scale reaction and do that on a robotic system. And the very nice thing about it is you can transfer that directly onto a plate of some sort of antimicrobial assay and detect that directly and find out which one of the compounds is the lead. So that's pretty much what that study's on. It should come out very soon.
Gentleman at the back.
Well, you mentioned it yourself, so I mean, I just wanted to say a bit more about it. You know, if you get a hit to a biologic in your cell-free system,
yeah.
Then when you move to working with bugs, obviously you've got a membrane that's getting
yeah.
between your peptide. So how do you get around that? Because a lot of antimicrobial peptides are membrane disruptors, aren't they? That's it. So,
how do I get around that?
how does one get around that?
How does one get around that? I think this particular system is useful for finding just initial hits, okay, and then they can be characterised further in terms of biochemical assays and in vivo assays. So I think it's about. I don't think there's enough evidence that suggests there's any sort of themes between one peptide and another, and why that would actually facilitate transport across the membrane. I think it's an interesting area to look at, really.
We've actually tried that. We did a cell-free extract protein synthesis assay, screening already active molecules. So to find which of that large pool of active molecules was protein synthesis inhibitors. So that helped us to narrow the field.
Sorry. Scalability of this thing. I mean, eventually you could probably go on the way of refining the production, shrinking it down.
I don't think the advantage of this is making. I think there's potential making natural products with just enzymes in a one-pot reaction. I don't necessarily see an advantage of using this for making a particular compound with cell-free protein synthesis. I don't think that's its strength. No.
Okay.
Personal opinion, but I may be wrong.
No.
Sorry.
No.
If I don't say Matt, it won't invite me.
I think I agree for high throughput screening. It's ideal, and we're definitely interested in this. Is there an upper limit on the size of molecules you can make?
I've made up to. So I've used it for a couple of different things. So I'm mainly interested in certain biosynthetic pathways. I haven't demonstrated any of the antimicrobial activity. It's very much something I'm looking at the minute. But I've looked at an NLPS enzyme. I've gone up to 180 kilodaltons using a Streptomyces cell extract. So that's a single module NLPS. I've looked at 25 biosynthetic enzymes from Streptomyces, and I can get 24 of those synthesized, and most of those you can see those on the S-page gel. So it is quite robust. So I mainly work with the Streptomyces cell-free system. You can pretty much use any given organism and make an extract and study that.
Sam, very quickly.
Yeah, I was just going to ask what your kind of success rate is with trying to use these pathways in a cell-free system.
Very limited.
Okay.
I saw a study of someone screening hundreds of clusters and trying to put it in vivo, and also that was also very limited. I've looked at these are the 4 examples I've tried. Three of them work, one of them doesn't. So the two top ones we knew the product of the reaction. The bottom one we suspect we know it, and the third one we don't actually know what the product's going to be. So we've done it's a very limited proof of concept showing that we can get it to work.
Okay, thanks.
Can I invite Michael Voss? Thank you. Did we give him a round of applause? Give him a number.
All right, thanks a lot. Very interesting meeting, very diverse talk, so much so that I am developing a mild case of imposter syndrome, as I'm a simple microbial ecologist, and I've only done one study touching on natural product discovery, and that's what I'm talking about with a very talented undergraduate student, Amy Cole-Clough, who's now doing a PhD in Birmingham. I'm even more embarrassed to say that it started off as an excuse to go rock pooling in the boss this time. But it was a very interesting project besides that. So it led to a publication which you can't read in Evolutionary Applications because of Abby's hard work. So what we did on the shore was looking for seaweeds, which are known as very prolific antimicrobial producers, lots of different secondary metabolites. There's hundreds and hundreds of papers where people pair different extracts with viruses or with bacteria or with protozoa or with human cell lines, and there's lots of activity, probably because seaweeds are in a soup of bacteria and they don't want any fouling, so they might produce that. But there's lots of different interactions going on between microorganisms and seaweeds. There's also beneficial interactions, and there's lots of interesting aspects to it. Our study was relatively crude, but for instance, seaweeds produce different antimicrobials depending on location or disease status, or even like the part of the plant, if it's the front or the holdfast. There's different expression. So a lot of these studies, I think the term is grind and find, where you just, okay, so that rings a bell. And we thought we'd do it in a little bit more of a systematic way. So I'll just talk you through. So that's me scuba diving again in the boss's time. So that was fun, and we collected 48 seaweed species and put them in the freeze dryer. Then used the James Martin spice grinder, a valuable lab tool, and end up with these powders that we made methanol extractions of, and then we dip these discs, and you get all these nice colorful discs that you let the methanol evaporate off. And then we got a collection of Staph aureus isolates from the local hospital, Royal Cornwall Hospital. So these are all isolates from actual human infections. There are 28 different ones based on their Phytec profile. So I don't know if you know Phytec, so that's kind of an incubator with 20 different kind of antibiotics to give, like the MICs. So we knew that we were all distinct. And so you see, so we got a whole lot of data with these different kind of inhibition zones with these 48 seaweed species times 28 Staph aureus isolates. So, as we suspected, seaweeds are a promising source of antimicrobials, so there's kind of a lot of evolutionary divergence between seaweeds. There's red ones and green ones and brown ones, so they all had activity. About half showed anti-staph activity. Most of them showed activity against the whole panel, and then a subset, I think it was 17 or less, showed activity against only a subset of strains. I guess the problem was, so this plot here, where so every dot represents one of our strains. We plotted the clinical resistance, so the number of clinical antibiotics the strain was resistant to against the number of seaweeds extracts, crude extracts, of course, containing hundreds of thousands of different types of molecules. And you see that there's a positive relationship, which I guess is bad news for natural product discovery. There's a cross-resistance generally, so if things are just more resistant to antibiotics, they're also more resistant to new antibiotics and antimicrobials. And then we took a more kind of up-close look by looking at the actual size of these inhibition zones for different seaweeds, and also inhibition zones caused by different antibiotics. And it was a clever idea by Abby, so she plotted the inhibition zone size for a given extract against inhibition zone size across the panel for a different antibiotic. So if you see a kind of a blue dot, so you can't read it, sorry, but so these, you have to believe me that these are seaweeds, and in bold there's antibiotics. They're plotted against each other, so if you see a blue spot, there's actually a positive correlation, and if it's bluer and larger, it's a stronger correlation. Red, there's a negative correlation. So the blue is kind of cross-resistant, so things that are more resistant to the antibiotic are also more resistant to the extract. But red is something called collateral sensitivity, which is basically what you want. So you want to have a new compound that's actually active, especially against resistant things. So that kind of pointed at the activity of promising, potentially promising sources. So I thought that was a quite nice, kind of more systematic way of looking at the activity. And we looked at a couple of other things. So there's lots of invasive seaweed species. You might know of the sargassum that we have here on the shore. They don't have more killing activity. We also looked at some of the seaweeds came from the same genus, so these are all the same genus, and they didn't all have the same activity. So you can see that there's one. This one is an outlier, so the more red, the greater the inhibition zones across the panel, which is actually interesting because the one in the middle is genetically the most divergent strain, and it's also most divergent if you look at the activity against gram negatives. So it's not that if the kind of the genetic divergence or the activity on a gram-negative, for instance, does not explain what you will find if you look at a gram-positive. So it's good to keep an open mind if you screen your organisms. So I guess really briefly, I guess it's very easy to find antimicrobials, at least these fruit extracts, so that's good news. And even closely related species produce different compounds. This is bad news, but if you look up close, you can. There's ways to detect things like this collateral sensitivity that allow you to look at potentially more interesting compounds. So it's a bit more evolutionary ecology informed natural product discovery. I think it brought kind of up an interesting question to me, and I don't know if there's any truth in it, but you can imagine that if things are settling on seaweeds and are resistant to the seaweed, and that makes them also more resistant to clinical antibiotics, there might be a potential for kind of co-selection by, which we know of course from soil microorganisms, because that's how antibiotics and antibiotic resistance evolved hundreds of millions of years ago. But potentially that's also the case for kind of macroorganisms. It would be interesting to know what you think about that. I couldn't find much in the literature about that. So you could imagine that if there's, for instance, Staphylococcus aureus, which survives pretty well in seawater, if they form biofilms on the kelp, who knows, they could develop resistance through point mutation or either lateral gene transfer with a kind of a native microbiota. So bottlenecks, of course, we then thought, well, we have all these interesting compounds, let's see what it is. And that's kind of where very quickly hit the limits of my knowledge. It's kind of a really different sport to look at all this stuff. But I worked a little bit with Ruth Ayres, who was at the PML, and we did some work. We got a little bit of money, but it was really not enough money, not enough time to really get somewhere. But the approach we did was fractionate or degrade or different compounds to see if it lost activity, to then in the end do kind of comparative metabolomics and compare where peaks have changed to indicate that these peaks are representing kind of the active compound. But that was quite difficult. I've had some industry contacts, but I also quite find it difficult to extract money from them. So companies tend to be careful with money for some reason, successful companies in any case. We did some sequencing of our Staph aureus isolates for kind of genome-wide association studies. I don't know if you know about that, to kind of see if differences, if algorithms could predict kind of what genes were responsible for which kind of resistance pattern. It was also very tricky. So it was a lot of work, and in the end I just got like one little tree out of that. And we've also been looking at bacteria from seaweeds, but I guess we can talk about that later. And I'll keep it here. Thanks.
Thank you.
I just want to end with this slide. This is my favorite seaweed. It's a bushy rainbow wrack. It's iridescent, so it's a beautiful kind of blue-purple color if you look one way, but if you look at a different way or above water, it's brown. It's a bit like if you look at a CD or at like oil slick. And I thought this was a nice quote, and that's not how we write in journals anymore, but I thought it was quite entertaining. Anyway.
But is it antimicrobial?
That one, yeah, had antimicrobial activity. Yeah.
Great. Any questions for Mikhail? Gary, hand up first.
So, different kinds of algae, red, green, brown algae.
Yeah.
You can use that. I think brown algae use a lot of Toxicity species for aroma for oxidation, so they're generally quite bacterial free, but they're not totally bacterial free. But anyway, the thing I wanted to tell you was there's a project going on for Roscoff right now, where they're sequencing 40 brown algae genomes.
Okay.
So it might be interesting to speak to the guys,
Yeah.
look at the organisms that they're using, and then you might get some genetic insight into what resistant mechanisms are. Second thing, did you really control the first samples, like test them for toxicity against anything else?
What do you mean?
Well, you
I mean controls as in like methanol, like the act.
No, I mean, did you test them, for example, against cell lines or?
Oh no. So of course, you know that would kind of maybe. Actually, she played around with some Galeria assays, but that's not in the paper. So probably a lot of these things are actually also bad for us. So yeah.
Any of them edible seaweeds? Because if they're edible, that kind of answers the question.
Yeah. There's about 10 or so.
10 are edible.
Yeah.
I think looking at the diversity of microbes, in the context of 4 different
Yeah.
Just wondering, one, what sort of biomass do you use? How much seaweed do you use for your spice grinding things to get your extract? Do you have to process a lot of seaweed? And do you know how your seaweed microbiota relates to the killing spectra? And the
Yeah, that's a good
production
question.
of the-
So we kind of rinsed, of course, the seaweed and cut off necrotic bits, but of course the original kind of the bacteria were still on there. So it could be potentially that the activity was kind of mediated by the microbiome rather than the seaweed itself. I don't think, because the biomass is so much lower than the actual seaweed, I don't think that's probably the case, but we can't exclude it. Yeah.
So I've got a comment and then a question. So the comment is, I don't know if you're familiar with the grant.
Probably not.
Right, Moore's he's just got some eye-watering sum of money from the Veterinary Institute to sequence some hundreds of seaweed genomes, and the plan there is to basically establish the ontology of the seaweed, setting the metabolism. So
Okay.
we're having a look at that.
Yeah, definitely.
The second question, which actually is not related to antibiotics, but this kind of iridescent property, does anybody know what the kind of physiochemical basis of that is? And if they do,
Yes, someone at Bristol actually.
Right.
Heather Whitney. Yeah, I actually showed her this thing because she gave a talk about plants, and I said, "Do you know about this? My favorite seaweed?" And she didn't. I showed her, and then she wrote a nice paper on it. Yeah.
Good for her. Have you thought about exploiting that as an adaptive camouflage? So there's an awful lot of interest and funding available at the moment to have certain routes for bio-inspired adaptive camouflages and missile trains.
Yeah.
That's a good thing, right?
But you will be camouflaged against very blue things. I don't know how many.
Last question from the back, please.
Very practical question. Are you drying your seaweed before you grind it, and have you tried different temperatures and durations?
No, we just kind of shoved it straight into the freeze dryer.
Okay, so you
Yeah.
freeze dry it.
Freeze
and then you
dryer.
grind it.
Yeah.